MSS, should we start, sir?
Yeah, I should. Let us wait for another 5 minutes. Huh?
Okay, sir.
Yeah. Dr. Jaiprakash. Shilpa.
Sir? Sir?
Yeah, we can start now.
Oh, okay. A very good morning to all of you. I present my humble pranams to the lotus feet of His Holiness, and I welcome all of you for the webinar on vaccine safety, monitoring and research, organized by Regional Training Center for South Zone, PBI, Department of Clinical Pharmacy, JSS Medical College and Hospital, JSS Mysore. I request now Dr. M. Ramesh, Coordinator, AMC, JSS MCH, to kindly welcome the participants.
Thank you, Shilpa. A very good morning to you all. Seeking the blessings of His Holiness, I take this opportunity to welcome the guest, eminent speakers, and all the participants for this webinar on vaccine safety monitoring and research, organized by the Regional Training Center for South Zone, JSS Medical College and the Hospital, Mysuru, in association with the Pharmacovigilance Program of India, Indian Pharmacopoeia Commission, Ghaziabad. The Regional Training Center located at JSS Medical College and the Hospital, Mysuru, is regularly conducting training sessions for the benefit of technical associates and coordinators, and also for the healthcare professionals and health profession students. It has taken several initiatives, not only to create awareness on importance of patient safety and reporting culture, but also to promote the safe and quality use of medicines and vaccines. As patient safety is paramount concern for healthcare professionals following vaccination, this webinar is being held with an objective to provide an insight into the essentials of vaccine safety, including its concept, surveillance, research, and decision-making process. And we sincerely hope that this webinar would enable the participants in conceptualizing the concept and importance of vaccine safety surveillance. With this brief note, I am honored and privileged to welcome all the guest expert speakers and participants of today's webinar. Firstly, I extend a very warm welcome to Dr. Jay Prakash, Officer in Charge, Pharmacovigilance Program of India, and Senior Principal Scientific Officer, Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, out of India. Dr. Jay Prakash has achieved several milestones in his career, and his contribution to the current Pharmacovigilance Program of India is immense and laudable. Sir, we are very privileged to have you with us on this occasion. We welcome you, sir. We are supposed to have with us Dr. T.M. Pramod Kumar, Principal, JSS College of Pharmacy, Mysuru, but because of his other schedule, he could not make it for today's program. But however, he has conveyed his best wishes, and I welcome him in his absence here. Further, I extend my very warm welcome to Dr. Madhavaram Balakrishnan, Medical Officer, Global Vaccine Safety, Pharmacovigilance, World Health Organization, Geneva, Switzerland. Dr. Madhava is a medical epidemiologist, an international scientist, and an expert in vaccine safety. He pioneered the WHO AAFI causality assessment methodology and the development of several tools, trainings, and software for vaccine safety. We are very glad and delighted to have Dr. Madhavaram Balakrishnan with us for today's webinar as one of the eminent speakers, and we welcome you, sir. Also extend a very warm welcome to Dr. M.D. Ravi, Professor, Department of Pediatrics, JSS Medical College, JSS HR, Mysuru. Dr. M.D. Ravi has vast and rich experience in teaching, practice, and research that spans over 35 years. He served the institution in different capacities, including Vice Principal and Director of JSS Hospital, Mysuru. He is a renowned person in the field of vaccine safety and research. It is a great honor to have Dr. M.D. Ravi with us today as one of the expert speakers. We welcome you, sir. Further, I extend my heartfelt welcome to Dr. Meera Desai, Professor, Department of Pharmacology, Newton Medical College and Research Center, Visnagar. Dr. Meera Desai is a clinical pharmacologist with more than 30 years of experience in medical education, clinical research, and drug safety monitoring. Madam serves as expert member in several committees of various organizations, including PVPI and CDSCO. We are very glad and honored to have Dr. Meera Desai as one of the eminent speakers on this occasion. We welcome you, madam. Also, I extend my hearty welcome to Dr. K.B. Chetak, Assistant Professor, Department of Pediatrics, JSS Medical College, Mysuru. Dr. Chetak is a nodal officer for COVID-19 vaccination program at JSS Hospital, Mysuru. And vaccination is one of his research areas of interest. We are very glad to have Dr. Chetan as one of the speakers for today's webinar, and we welcome you, sir. Further, we have with us Dr. Juni Sebastian, Assistant Professor, Department of Pharmacy Practice, JSSCP, Mysuru. Dr. Juni is a pediatric pharmacist and involved in coordinating the vaccine safety surveillance at JSS Hospital, Mysuru. We are very happy to have Dr. Juni on this occasion as a speaker, and I extend a very warm welcome to Dr. Juni Sebastian. We welcome you, Juni. Also, I welcome Mr. Shilpa Palaksha, Associate Professor and as Biology Lecturer, Department of Pharmacy Practice, JSS College of Pharmacy, JSS HR, Mysuru. Last but not the least, I extend my heartfelt and very warm welcome to all the participants who have taken time out of their busy schedule to attend this webinar. I welcome you all. Finally, I welcome one and all. Thank you very much, and over to you, Shilpa.
Thank you very much, sir. On behalf of the team, I also welcome you for the webinar, sir. Now, I request Dr. Jay Prakash, sir, Senior Principal Scientific Officer and Officer in Charge, PVPI IPC Ghaziabad, to address the participant. Jay Prakash, sir.
Dr. Jaiprakash. Phone switch off. Shilpa,
Sir?
yeah, he has lost the connectivity. I think he is reconnecting now.
Okay,
Give me-
okay, sir.
yeah, that will be a minute.
Okay, we'll just wait for a minute.
Yeah, for a minute.
Jaiprakash, sir, can you hear us, sir?
Jaiprakash?
Whether I am audible to you or not, I also.
No.
I think it's not noted, but I don't know what is the problem.
Your video is also not appearing, Jaiprakash.
So I am. It is already on. It is not visible.
Yeah.
You please put it on from your side, sir.
Yeah. I just can you speak over the phone? I just put it on the speaker.
Hello.
Hello.
Am I audible now, sir?
No, Jaiprakash.
So I am getting some problem, sir.
Shilpa,
Sir?
can you hear his voice over phone?
Very low voice, sir.
Low voice.
Yeah.
I just put my speaker on and I have picked up, sir.
His mic is switched off.
Will it be all right?
He has to unmute now. Okay, now he unmuted.
Vice Chancellor, DSO Secretary of Higher Education and Research, Dr. T.N. Pramod Kumar, Dean, Faculty of Pharmacy, DSO Secretary of Higher Education and Research, Professor M. Ramesh, Coordinator of this PNC and Regional Training Center, SPC, and my dear participants, I feel honored to be part of this webinar on vaccine safety monitoring and research, and I congratulate Professor M. Ramesh for organizing this important webinar for the interest of all the participants. Research body of pharmacy has been one of the most active AMC as well as the regional training centers under the pharmacovigilance program of India, and I request them to keep such activities up. As you all know, that India is one of the major exporters of the vaccine, and it supplies vaccines to more than 150 countries of the world. Safety and efficacy of vaccines is of paramount importance. You know, over the last 2 years, the emergency measures have also been taken by the Central Drugs Standard Control Organization during the COVID-19 pandemic to give the emergency authorization for many of the COVID-19 vaccines. And these vaccines are the life-saving drugs, and under the New Drugs and Clinical Trials Rules 2019, vaccines are always considered as the new drugs. As you will be knowing that in the pharmacovigilance program of India database, there are around 6 lakh of the individual cases reports. Out of them, more than 36,000 ICSRs are of vaccines alone. If you look at the COVID-19 vaccine, the majority of the vaccines which have been COVID-19 pandemic are the coincidental as well as the vaccines. And these vaccines have been used mainly in the age group of 18 to 44. And we have not seen any serious adverse events in the COVID-19 vaccine. Just the mild adverse effects, fever, pain at the site of the injection, nothing weakness. These mild type of the adverse effects for the PPI. And therefore, these vaccines are quite safe and have saved the life of the majority of the population of the country. The quality standards of the vaccines are also there in the Indian Pharmacopoeia, the IPC, and more than 70 vaccines quality standards are there in the Indian Pharmacopoeia. We are in the process of bringing out the next edition of the Indian Pharmacopoeia. So, I have seen the program and it has been very well designed, and we have the from different parts of the country, and few of these speakers, like Professor Meera Desai, is already on the panel of the PPI, not only for technical assignment, but as well to give any sort of the work that to provide her services to the pharmacovigilance program. So many experts have been supporting for the national board of pharmacovigilance, and I request the organizers to organize such meetings frequently of the participants. And with this, I congratulate again the team of Dr. Ramesh for organizing this beautiful event, and participants will shortly benefit and have the effective question answers during the webinar and the effective benefits. So with this, I again congratulate the organizers for the success of this program. Thank you very much.
Thank you, Dr. Jayaprakash. Shilpa?
Yes, sir.
Then-
Thank you. Thank you, sir. Thank you, Dr. Jayaprakash, sir. Jayaprakash, sir, has conveyed his message, and he has highlighted the importance of today's webinar. So with this, that he has passed his greeting to us. Now we will move on to our scientific speakers. The first speaker of today's webinar is Dr. M.D. Ravi, Professor, Department of Pediatrics, JSS Medical College and Hospital, Mysuru. M.D. Ravi, sir, has been working in this area for a long time. He has a rich experience. He has a teaching experience of more than 35 years. He has served in various capacities at the hospital and also in the college. He has conducted more than 20 national and global clinical trials at different phases, which even included the WHO multi-country collaborative vaccine safety study. He has trained to be a data safety monitoring board member from Harvard University in 2013, and he has served a member for 3 vaccine trials. He has several publications and many of them scientific presentations to his credit. He is a renowned speaker and has received many awards to his credit, including the Seva Ratna Award from Mysore Circle and the Best Teacher Award from JSCHER. And he's also been a visiting professor at AIMST University, Malaysia. With this brief introduction, I request Dr. M.D. Ravi, sir, to kindly take over and deliver the lecture.
Good morning, everybody. Welcome to the webinar, and thank you very much, Shilpa, for that introduction. So we'll go straight into the program. Okay, so you can see the screen.
So we can see a slide, sir.
Yeah, thank you. Right, now, what we are going to do is just do a brief introduction into what on immunization safety. You know that there has been a lot of
Ravi sir, can you put it on the slideshow mode or presentation mode, sir, your screen?
It is on a slideshow mode. Let me just try it again. Is it okay now? Is it okay now?
We are viewing it as a slideshow only, sir. Slide is viewed, not as a presentation.
No, because here in my screen is coming as slideshow. I think there's a problem with your link. Whether it's come.
Okay, sir. Okay, sir. No problem, sir. You please continue.
Because in my thing, it is coming as a slideshow. Okay, anyway, it doesn't really matter. The problem is that this thing won't run. Your animations won't run.
Sir, could you please reshare it, sir? It will be all right.
Okay. Okay.
Yes, sir. Now it's fine.
Okay.
Thank you.
Now, we know that vaccination is one of the greatest things that has happened as far as disease prevention is concerned. Apart from safe water, vaccination has been proved to be the most effective method of preventing disease processes. So vaccination has been there for a very long time. Currently, it's estimated that we are saving somewhere between 2 to 3 million lives per year just on the basis of vaccine. So there is a lot of problems in some places with anti-vaccine lobbies. And these anti-vaccine lobbies are the ones who are waiting to find reasons for them to say that vaccination is unsafe. So what is the program all about today? One is, why should we make vaccines safe? Second is, how do we make vaccines safe? Third is, how do we assess vaccine safety? So this is what the program is all about. We know that there is things called antigens, which are what the infective organisms have. The body reacts to antigen. It doesn't really care where the antigen came from, whether it came from a natural infection or whether it came from a vaccine. The response will be the same. So it will produce antibodies, and most of the time, if you've got a good vaccine, it will produce what are called memory cells, which will go and sit inside the what's called a germinal center. And then the next time the antigen comes into the system, there is already a clone of cells which is ready to produce antibodies, and there's a very rapid response of the antibodies. We know that natural infections in general, not all, in general produce better immunity than vaccines and maybe longer lasting in some cases. However, the cost involved, I'm not talking the cost in terms of money, but the cost in terms of morbidity of getting immunity due to a natural infection is far, far higher than that due to a vaccine. Morbidity due to natural diseases, for example, measles, the risk of death, the risk of severe complications, hemorrhagic measles, or post-measles complications such as bronchopneumonia, bearing up of tuberculosis. This is the cost involved. Whereas the measles vaccine per se is a very safe vaccine and it produces pretty good antibody levels. There are some natural infections which don't produce good antibody levels. For example, typhoid in children doesn't produce very good antibody levels. There, the chances of relapse are higher and the vaccine will prevent their relapse. So, morbidity due to natural infection is almost always significantly higher than due to a vaccine. So, if you want to say you make the vaccine administration part safe, it's not very difficult. You have to follow some basic principles of using the vaccine, just like giving any other injection, hand hygiene, proper sterile technique, and importantly, you have to ensure that you're giving the proper vaccine. So using a diluent, use the proper diluent. If you are using a particular vaccine for a particular age group, then you have to check the dose, you have to check the route, and commonly we monitor these children or patients for about 30 minutes after the administration of the vaccines for immediate side effects. Most important of which is anaphylaxis. So if anaphylaxis is going to occur, it's going to occur within 30 to 40 minutes. So we are monitoring them for a short period of time. You have to be prepared to handle an adverse event if it happens. Because if an adverse event happens and you are not able to handle it, you may lose the patient, and that will be a very major setback for your vaccination schedule, apart from the fact that it's a disaster. Now, especially when you are dealing with vaccines which are coming from elsewhere, you need to make sure that the transportation has been proper, there has been a cold chain, you have to check the temperature that's been maintained, the storage of the vaccine in your site has been proper, the transport from the storage department to the place where it's actually administered. All this requires to be checked so that you do not have vaccine degradation and you do not have problems due to the vaccine administration. A simple example, we know now that we do not give vaccines into the buttock because there's a chance of sciatic nerve injury, right? If you give it into the anterolateral thigh or into the deltoid, your chances of injury are almost nothing. So proper route, proper location, spending a little time to decide how to give the vaccine and where to give the vaccine is important. It's also important because some vaccines which are given into subcutaneous fat, like the rabies vaccine, do not work at all. So, why are we so bothered about vaccine safety? We have drugs which are giving, we are giving vaccines. The difference is that drugs are given to people who are unwell. So, some amount of side effects may be tolerated. You give, let us say, an anti-inflammatory, the patient has some degree of gastritis, even tolerated because the pain of the arthritis is worse than the pain of the gastritis. We will accept it. But vaccines are given to children who are well, and these are preventive. So the tolerance for a side effect will be significantly lower in a child who is getting a vaccine than in an adult who is getting a medicine for, say, an arthritis. So because of the lower risk tolerance, we have to be careful about looking for uncommon or rare side effects. This is the reason why we have this surveillance system, so that we can identify problems which were not identified earlier. If there has been one adverse event which has become publicized, then it can derail an entire vaccination program. Some of you probably know about the disaster that happened with the HPV part study, I think it was called in Andhra Pradesh, where a couple of children died because of the HPV. It was thought it was due to the HPV vaccine. However, subsequently it was found it was not due to the HPV vaccine. But because of that, the whole HPV program got delayed by several years. So, one significant adverse event is enough to delete an entire program. We've all heard about the problem we had with the pulse polio program some years ago, when there was a rumor that some children died after the pulse polio, and then there was a big hoo-ha about it. In our hospital itself, we had about 400 or 500 parents coming in the night between 10 and 1 in the morning. We were all here dispelling myths about this kind of stuff. So adverse events are something that can cause a major kind of community problem. So if you have a proper surveillance system and you have the ability to understand what are the adverse events and take care of them adequately, then this kind of problem will not happen. That means essentially you must understand the disease, you must understand the vaccine, and you must know what are the side effects that you're going to look for. Now, any vaccine is approved only after a lot of efficacy and safety data is reviewed from studies, controlled, randomized clinical trials. You will be hearing about what are the types of clinical trials probably later on, or maybe you'll be reading about it. But any vaccine goes through a very rigorous testing before it is allowed to be put into the general population. So a lot of safety checks are there. But after the approval, vaccines are used in the real world. And here we are giving it to a very large population as opposed to what we have done in clinical trials. So it's possible that you will get adverse events which were not picked up in the clinical trials. Therefore, an ongoing surveillance for adverse events is something that's very important, so that we can identify newer problems that are occurring with any given vaccine. So this is necessary for the development of a sound policy, national level policy, or a programmatic policy, let us say from the Indian Academy of Pediatrics or the Indian Medical Council or whatever, and recommendations. So this surveillance is the thing that's going to give you all the information that you require to make or modify the existing policy. Now, you have to look at policies in two ways. There is a vaccine policy which is state, which is a national policy, and there is a vaccine policy which is for an individual patient who is coming to a private clinic. There is a little bit of a difference in how we are going to look at it. For individual, for a state policy, it is important to look at the cost, it's important to look at the herd immunity, it's important to look at the benefit to the community. And in an individual patient, you will be looking more at the efficacy of the vaccine. In both cases, you will have to look very carefully at the safety of the vaccine. But in an individual place, you may not look very much into the cost of the vaccine. So you may go for a higher cost vaccine, which has got a greater margin of safety. Whereas in the state policy, you may go for a slightly lower cost vaccine, which is safe, but the margin of safety may be marginally or fractionally lower. So there is a difference in how we are going to assess the requirements for a vaccine program. An adverse event is more likely to impact the state program rather than an individual decision as to what vaccine you're going to take. So when you get across to an adverse event, you need to classify it. That is something that will be talked about later. Then you have to decide whether the vaccine was the culprit for this adverse event, or this is called a causality assessment. If this is not properly done, you will get a whole lot of backlash, which is not necessary. As an example, one particular vaccine, the one which I told you about HPV, the deaths were subsequently found to be not related to the vaccine. However, the information had already been spread that the vaccine caused deaths, and therefore the vaccine program got delayed. Sometimes it is a delay on our part in dispelling a myth that causes a problem. As an example, that around the same time there was a death in, if I remember right, in the UK following HPV vaccine, but within a short period of time, the government came out and said that the death was due to a mediastinal tumor and not due to the vaccine. So the vaccine program did not get delayed. So we need to do the causality assessment and make it public as fast as possible. Without a proper classification and a proper causality assessment, we cannot do an adequate corrective and preventive action to prevent this kind of event from happening again. Now, it's not necessary that only serious adverse events in the sense that the patient has got into a hospital or died. It's also now a serious adverse event can also be a community impact that the population at large has become very worried about this particular vaccine, and this needs to be addressed. However, this has to be handled diplomatically, and whatever facts are there, they should not be falsified, because if it is falsified and then it comes out that it is falsified, the damage done is more or less irreparable. So, involved health worker has to be therefore aware of the vaccine, its effects and its side effects. During the process of vaccination, proper documentation is essential, including the storage that you've done, the administration, whether you have talked to the patient, given him all the details about the vaccine, and he has accepted to take the vaccine, he's been told about possible side effects. If the documentation is proper, then there is no problem at all. All these will be required, all this data will be required in case a major adverse event occurs. Minor adverse events, probably nothing is going to happen, but if there is a serious adverse event, then there will be an investigation and all this data will be required. It's not only the effect of an adverse event on the patient that's important, it's also the effect on the community. If the effect is there on the community, the sense is that the community gets worried about this particular vaccine, then that itself is going to be something like a serious adverse event. So proper safety assessment, if you have done a proper safety assessment and you have proper surveillance in place, you can dispel myths which have occurred because of this long information or because of this panic that has happened with the general population. Knowledge, therefore, about the vaccine and about its adverse events benefits both the healthcare worker and the recipient. If you are knowledgeable about side effects and you can explain to the recipient that, look, this is the side effect that you had, this is because, this is something that happens with the vaccine, it's not going to cause any problem, so he's likely to continue with the vaccination program. And at the same time, you have not lost your credibility. So it's important that the person who's doing the vaccination should be aware of everything about that particular vaccine. So there is an old idea, if you are forewarned, if you know about it in advance, it is forearmed. And this is something that holds true even now. Thank you very much, and I will hand over to the rest to give you greater detail about vaccines. Thank you very much.
Thank you very much, sir. We'll take up the questions in the last session, sir.
Yeah.
Now I request our second speaker, Dr. Jimmy Sebastian. Dr. Jimmy Sebastian is working as an assistant professor at the Department of Pharmacy Practice, Jeffrey College of Pharmacy, Hospital. Julie is been especially working in the area of vaccine, and she has been trained. She has visited countries like Korea for her extensive training in the field of vaccine and immunization. She has been presented a number of papers, both in international platform and the national platform. She also has published many articles related to her expertise work in both national and international journals. With this small introduction, Jimmy, I request you to take the platform and deliver your.
Ma'am, hope the screen is visible and I'm audible for.
Yeah. You're visible. I'm audible.
Thank you, ma'am, for the introduction. So for the next 30 minutes, we are going to discuss on the monitoring and reporting of adverse events following immunization. So the presentation outline goes like this. So I'll be briefly discussing on the difference between safety surveillance of drugs and vaccines. Then quickly, what is the need of pharmacovigilance? A small introduction on adverse events following immunization and its types. Then we'll discuss more on the monitoring and reporting of adverse events, and I'll be concluding my session. So let us see what exactly the difference between the drugs and vaccines. So the vaccines are generally given for healthy human population and to a larger population at once. Many a time we are having the mass immunization campaigns for giving the vaccinations. But when it comes to drugs, the treatment is for ill patients and not to everybody. There is no large or a mass campaigns for drugs. We will be giving to a smaller population who are diagnosed to have that particular disease. And when it comes to drugs, there are only limited number of products, but there are a large number of products and many different classes of drugs are available for current use. And with a single dose, and some vaccines are available as a single dose only, there is a greater potential for temporal coincidence in case of vaccines. But these kind of coincidental event following a single dose is not commonly seen with the drugs. And coming to the vaccination programs, as we are giving it as a mass campaign programs and such things, there is many healthcare professionals are involved and many centers are involved, and the centers having a different capacity, or there may be a primary health center, secondary care centers, and sometimes it is through the schools, Anganwadi. So various levels of facilities are involved in the administration of vaccines. Hence, there is a higher chance of program errors and injection reactions and such things. But the administration related issues with a drug is not so commonly seen because it is generally given in the hospitals and may not be commonly seen. And vaccines are biological products, and there is a greater potential for lot to lot variations, and cold chain maintenance is very essential in case of vaccines. But when it comes to drugs, these are chemical products and storage and handling is not so critical like a vaccine. And as I said before, the vaccines are given as mass campaigns, and many doses of vaccines will be given in a short period of time to a definite population. Many a time it will be given to the extremes of age. We will give to largely most of the vaccines are given for children, and some are meant for even adults or even all population, but greatly it is given to the defined age population. But when it comes to drugs, very rarely we are having mass campaigns, and to a very defined population we are giving instead of giving to a larger population or extremes of age. So when it comes to the vaccination programs, universal vaccination program is recommended, or otherwise we can say it is mandated for various reasons. So nowadays the vaccination program or basic vaccinations are required for getting a school admissions, or now we know we have a COVID-19 pandemic came in, then the COVID-19 vaccination is mandatory for a travel. So likewise, this is recommended or mandated. So the people are having some kind of hesitation for these kind of mandatory things. Then risk-benefit ratio of vaccine is different from drugs, because as I said before, we have explained the difference between the drugs and vaccine. So obviously the risk benefit assessment or ratio is different. Then pre-licensure studies, that is the clinical trial from phase 1 to phase 3, is not enough to assess the complete safety profile of a drug. Suppose when you are taking up a clinical trial of a vaccine, we will give 1 dose of vaccine. Then the follow-up period for the recruited patients is just 28 days. So after 1 month of follow-up, the study is over. And if the vaccine is it is a single dose vaccine, then the study will go from the time of enrollment. The study is getting over after 28 days of follow-up. So the short period of time we are following, and we cannot identify something which is delayed, or we are recruiting only small number of population. Throughout the clinical trial, we will be recruiting somewhere around less than 4,000 population from phase 1 to phase 3. So a very small number of population is enrolled. And while enrolling, we are having a very definite inclusion-exclusion criteria. So this criteria generally avoid people with some kind of disabilities, people with diseases. So many inclusion-exclusion criteria we have. So we are taking a very controlled population and we are trying to identify the safety and efficacy. But that is not the case when the vaccine is introduced into the common in the market, or it is given for the population. So so the safety data, whatever we are generating from the pre-licensure studies, are not enough. And as we are recruiting only less number of population, the rare events we cannot identify. During the clinical trial, we do clinical trials only for one vaccine, and we do not give multiple vaccines during the clinical trial. But when it comes to the market, or once it is available in the national immunization program, it is rare that a person will receive one dose. There is a possibility of receiving multiple vaccines together. So that situation is not actually studied during the clinical trial. And whenever there is a pandemic or whenever there is an epidemiological changes, the existing the vaccine is of no use. So you may have to go with another vaccine, which is covering all these genetically mutated strains. So there is a possibility of, or there is a need of new, more and more vaccines, depending on the situation. Or in case of endemic or pandemic, we need to introduce more and more vaccines, and quickly we may have to release these vaccines. And obviously, as I said, it is mandated or it is recommended. The people are having a low tolerance on adverse events, because we already said the vaccines are generally given to healthy human volunteers or healthy population. Anybody who is having any acute disease or any chronic infectious disease or something, generally we do not recruit them for, or we will be giving the vaccination. So people, healthy population receiving a vaccine and ending up in having some kind of unwanted effects is not actually tolerated by the human population. And obviously, most of the vaccines are given smaller children, and receiving the vaccine following to that, the children is becoming ill is not tolerated by the parents. And the expectation, so because of all these things, expectation on the safety standard is higher when compared to the medicine for the sick people. So because of this reason, the National Regulatory Agency, they ensure the rigorous safety and efficacy as well as effectiveness of vaccine in various stages. In the preclinical trial, during the introduction, they will correctly evaluate whatever the safety and effectiveness data what we generated from the preclinical trial at the time of approval. Also, they even look for the post-marketing surveillance studies to make sure the vaccine which is available is safe and effective. So why the vaccination is becoming more and more important? So there are more diseases are becoming vaccine preventable nowadays. So we know our COVID, the COVID came, pandemic came, we were in lockdown, then the vaccination has come up. So many different vaccine preventable diseases are seen nowadays, but the previous diseases like polio, measles, all such things are not so prominent nowadays, and people are not aware of the burden of those kind of vaccine preventable disease. Because of the young population is not aware of the burden of the previously controlled vaccine preventable disease, they discuss more on the adverse events of vaccines. And obviously, as we said, during the clinical trial, we cannot identify all the adverse events. So there is a possibility of potential adverse events, which is called a signal during the administration process. And nowadays, the use of social media are increasing. So people are more aware on the vaccine safety. So maybe than the efficacy part or than the components of vaccine, the people are discussing more on the safety aspect of the newly introduced vaccines. And depending on the changes or a pandemic, endemic, all that, the national immunization programs will be keep adding the newer vaccinations or new population in the routine campaigns or coverage. And sometimes there will be specific vaccines, specially developed for the developing countries, or a quick development of new vaccines during the pandemic. So because of all these things, the safety monitoring or vaccine safety surveillance is very, very essential. So if you look at the examples of safety scar, so some even as Dr. M.T. Devi has previously explained about the HPV vaccines and pulse polio vaccines, the program, maybe it is occurring in 1 or 2 population, but because of the media attention and the scare which is causing the population, there will be a hesitancy, or it may clearly damage the entire immunization programs. So I will be giving you 2-3 examples of these kind of safety scars which was happened previously in the globe. So in 1976, there was in the United States, there was increased GB syndrome following H1N1 vaccination that led to the discontinuation of the entire campaign. So then if you look at the 1998, there is introduction of rotavirus vaccine. Following to that, there was increased case of intussusception. So that the vaccine was withdrawn by the manufacturer, and later on the studies have undergone. But until now, though we have a rotavirus vaccine in the government program, the intussusception is an adverse events of special interest following rotavirus vaccine. So that there is a scar. Once it happens, it will be there permanently with the vaccine. And in Egypt in 1993, there were 3 deaths labeled as post-DPT encephalopathy, which led to the discontinuation of the entire immunization program for 2 years in Egypt. So the impact is too huge. The death was 3 cases, but that entire, for the next 2 years, no vaccination programs happened in that country due to this kind of problems. So if you look at what is an adverse event or AFI, it is any untoward medical occurrence which follows immunization, which does not necessarily have a causal relationship with the usage of vaccine. Anything which is happening in vaccine can be considered, or any unwanted events which is following vaccination can be considered as an adverse event. So it may be an abnormal symptom, it may be an abnormal sign, abnormal lab finding like a thrombocytopenia or something, or it may be an abnormal disease, anything. Okay, it may be related, may not be related, but can be considered as an AFI, which is eligible for report. So if you clearly look at the definitions or types of adverse event, so there are 5 different types of adverse events, AFIs. One is vaccine product related reaction, where this AFI is caused or precipitated by the vaccine due to one or more of the inherent property of the vaccine. So it is happening because of the vaccine itself. It may be the antigen, it may be the additives, or it may be anything, but the vaccination product or vaccine product has caused the problem. The second type is vaccine quality defect related reaction. So this event or adverse event is precipitated by the quality defect occurring in the manufacturing process. So it may be due to the quality defect in the vaccine antigen, or it may be the devices which we are providing along with the vaccine for administration. So anything in the product or the administration device, some event is occurring due to a quality defect, which will be categorized under this particular category. Third one is immunization error related reaction. So this may be caused by an inappropriate vaccine handling, prescribing, or administration. So which includes there is some error happened during the transportation. So the transportation, we have to maintain the temperatures. The cold chain maintenance is very essential for vaccine transportation as well as storage. So if the vaccines are not transported or stored in the required temperature, there is a possibility of vaccination failure or loss of potency of vaccine or some kind of issues. Then handling, like while reconstitution, there is a possibility of error. While identifying a patient, there is a problem, there is a possibility of problem. Then while administering, the route can get changed, or while prescribing, we have given vaccination to a person who has some kind of contraindications, or while administering, we would have gone to a different route, any kind of issues. During the administration, maybe it may be a non-sterile injection, it may be a use of contaminated needle, it may be a wrong patient, anything and everything which is happening as an error will be coming under this immunization error related reaction. And the next one is anxiety related reaction. This is also a very important one. This is basically happening because of the anxiety, either due to the vaccination or to the injection. So put together anxiety to the immunization. So this generally do not happen with smaller children, but it generally happens with the adolescents and adults. So it may be a tachycardia, or it may be dizziness, it may be a pseudo seizures, or it may be vomiting, anything, anxiety-related reaction, dizziness, all that can be considered as an anxiety-related reaction. Then the last one is coincidental event. This is the most important thing I feel always. It may not be coming under any of these categories, not the product, not the quality, not the error, and not related to anxiety. But it occurred well within the time window for the development of adverse event. It might have developed the next day. But initially, we would have felt that it is because of the vaccination. But later, we will realize it is not nothing related to the vaccination. It is something other than the vaccination which has caused. But it happened well within the time window, and people might be feeling it is because of vaccination. So those kind of events can be classified as coincidental event. So let us see some examples. So for vaccine product related reaction, there is an extensive limb swelling following the DTP vaccine. This is basically happening because of the whole cell pertussis component of the DTP vaccine. So this kind of event, this is because of the pertussis, can be categorized as vaccine product related reaction. The next is vaccine quality defect related reaction. It may be the failure. Here I have given an incident. So it may be the failure of the manufacturer to completely inactivate a lot of inactivated polio vaccine, which lead to polio vaccine. So inactivated polio vaccine means the virus is killed and the vaccine is prepared. So in United States in 1955, in the inactivated polio vaccine was injected, and where the manufacturer failed to completely inactivate it. So there were some live component of polio virus was present in the vaccine, and the vaccine was administered to a 1,20,000 population, and 40,000 developed mild polio, 200 paralyzed, and 10 died. So this is one of the famous incident which can be categorized under the vaccine quality defect related reaction. Third one is error related reaction. So there is an abscess formation due to improper technique of BCG vaccination. So some vaccines, instead of giving in the intradermal, it might have given subcutaneous. Instead of giving subcutaneous, it is given intramuscular. So these kind of problems, or there is a change in the site of injection, can lead to these kind of errors. Then anxiety related reaction, it may be the vasovagal syncope in an adolescent following a vaccination. So we can see here the people are in a queue, the kids are in a queue for receiving the vaccine. And this child is receiving, and the next child is very anxious. There is a possibility of an anxiety related event, but that also, it may be either during vaccination, just before or after vaccination, but that also need to be reported and documented. And for example of coincidental event, so it may be a fever which occurred following a vaccination. But later, so initially we thought that the fever is because of the vaccination program. But later we realized that, or we identified a malarial parasite in the isolated blood of the patient. It may be that at the time of vaccination, the malarial parasite is in the incubation period in the patient's body. So that might have developed later following the vaccination. So these kind of events can be called as the coincidental events. So it is not only the vaccines, the components. There are various components present in the vaccine. There are antigenic components. That is that we know there will be live attenuated vaccines. There will be recombinant technology developed vaccines. There are various vaccines. So various antigens are present. So the antigens can produce the adverse event. Along with the antigens, there are some components which is added to enhance the immune process. So like adjuvants and protein conjugations, these things are added to increase the immune response. So those adjuvants and protein conjugations, etc., also can produce adverse events. And also, addition to the components added for enhancing immune response, there are another components which is added to the vaccines to reduce the contamination. So to prevent the contamination during manufacturing, transport, and storage, we generally add antibiotics, stabilizers, preservatives, etc. So these antibiotics, stabilizers, preservatives, which is added to these vaccines, also can produce some kind of adverse events. So for example, the measles vaccine generally has a trace amount, less than 25 microgram of neomycin present. So this neomycin is actually added during the vaccination preparation to prevent the bacterial contamination of the culture media. So it will be multiple times it is washed, cleaned, everything, but still less than 25 microgram of neomycin present in this vaccine. It doesn't cause any kind of therapeutic effect in the person's body, and it is very, it may not be, it is an inert kind of material. But later, if the person is having some kind of allergic reaction or non-allergic to neomycin, there is a possibility of some kind of event, maybe hypersensitivity and such things can happen with the vaccine. So anything which is added can have, maybe the vaccine is produced using egg yolk. Somebody might be hypersensitive to egg product, that they may develop some kind of event. So it is not only the antigenic component, the components added to enhance the immune response or reduce contamination also has potential to cause the adverse events. So to understand all these things, we need to have an effective surveillance system or effective reporting system. So how exactly the AFI surveillance system, an ideal AFI surveillance system should work is, there is a large number of population who received a vaccination and some of them develop adverse events. There is a possibility that the people who develop these the adverse events may not go back to the vaccinator. Some of them may go to the hospitals, some of them go to some clinics, some of them will meet some community healthcare workers. So whenever they reach any of these, any of the places, we need to have an effective surveillance network which capture all these events. Irrespective of the severity and seriousness nature, we need to capture all these events and need to be reported and further investigated. So these AFI reporting and the investigation is helpful to come to a conclusion whether there is a real a causal association between the vaccination and the development of the event. So when after doing the causality assessment, we can classify that the event might have a consistent causal association to immunization. So maybe a product-related reaction, or it may be an indeterminate causal association. That means we are not sure whether it is because of vaccination or not. We will always have a conflicting trend or conflicting information. We are unable to conclude that it is because of the vaccination program. So those kind of events comes under the indeterminate causal association to immunization, or it could be inconsistent causal association, or it will be a coincidental event. So if you look at the AFI surveillance components, the initial thing is a notification. So the notification generally done by the healthcare providers. So health system or a healthcare, the vaccinator, the nurses, the vaccinated people from the Anganwadi or wherever, the doctor, medical officer, whoever, whenever they see an adverse event, they have to notify to the higher authority that there is an adverse event occurred. Then after the notification, we have to have a case report form which need to be filled, and usually that need to be submitted by the person who is taking the medical officer, and that need to be submitted to the medical officer by the medical officer to the district immunization officer. Then there they will decide which all events need to be investigated or further processing is required. So some mild events they will be keeping with them, and that may not be investigated immediately. But all the serious events, it will be further investigated. And there are some field workers available in the district immunization office. Those field workers, they will visit the site where the person received the vaccination. Also, he will be visiting the place where the vaccinee is living. So to understand the background incidence of any event. So it may be to understand whether there is a coincidental possibility. So what they do, they will visit this immunization area, the area where the patient is living, and they collect the entire treatment history and the past medical history, everything. And they make a case investigation form in the district AFI committee. And it will be submitted to the state immunization committee. And later it goes to the national Causality Assessment Committee or the Immunization Technical Support Unit. Then the causality assessment happens by the National Committee. That is a systematic assessment of information which is provided by these reporters, and they will understand whether there is a relation between the event or not. And all the causality assessment, if the causality assessment is performed, the causality assessment report will be published in the Ministry of Health website for information to the general population. And there will be a feedback and response. It will be, so the feedback and response given to the community as well as to the health system and the media if the event has caused some kind of community concerns. So let us see what are the events to be reported. So obviously all serious adverse events need to be reported. If you want to say it is a serious adverse event, the event would have caused some hospitalization, there is a prolongation of hospital stay, or there is a death, permanent disability, then a life-threatening event. So if anything happens likewise, then we consider that as a serious event. So if there is a serious event, that need to be immediately reported. And if you are expecting a potential error, so generally the clusters occur, most commonly the clusters are happening because of the error. So when there is a cluster or there is an error related event, that need to be further reported and investigated. Then any event which is leading to a parental concern, a community concern also need to be investigated. Or any event which is of unexpected severity. That means we expect that the fever following vaccination is mild. Okay, but in reality, we are seeing it's not mild, it is moderate or very severe. The temperature is too high. So if there is an unexpected severity occurring, then that event need to be reported. And also an unexpected frequency. So we generally feel that this particular event will occur in 1% of population. But we are not seeing 1%, we are seeing a larger percentage. So that means it is an unexpected frequency. That events also need to be reported. And like adverse drug reaction or a pharmacovigilance, the reporter should not assess the causality. So the causality will be assessed in the National Center for better understanding. So the reporter, while reporting, they do not want to assess the causality while reporting. And some events, when there is an error suspected, so where there is a bacterial abscess, local reaction, high fever or sepsis, BCG lymphadenitis, or all these things, or there is a cluster of event, the or any error is suspected, that needs a special attention, because when there is an error, the error may get repeated. So we need to address the person who made the error, or we need to assess the system to rectify the errors. So that kind of problems need to be immediately, or that needs a special attention. And any event which is of unexplained cause, which is occurring after 30 minutes of vaccination, also needs a special attention. And any event, even if it is mild, but if it is causing a parental or community concerns, these events also needs a very, very special attention. Okay, now we are ready we understood what are the different events to be reported. Now let us see what are the core variables to be present in an AFI report. So if you look at the WHO website, they have clearly mentioned what are the core variables to report an adverse event. So there are 6 events they clearly mentioned. One is a record identifier, information on the patient, vaccine, event, and any other comments. So let us see what are the record identifier. So it is the date of the AFI report, place of vaccination, country and the location where it is reported, and a unique number which will be generally given by PVPI or the national agencies. Then coming to the patient, there is a patient identifier, maybe the details of the patient, date of birth, age at the time of onset, age group at onset, the sex, as well as the history of the event. Coming to the vaccine, which is a primary suspected vaccine, both the generic as well as brand name is required. And what are the other vaccines given prior to this AFI? What is the dose number? Then what is a batch number? What is the diluent used? What is the lot number of the vaccine? All this is mandatory to notify. Then coming to the event, the date and time of vaccination, date and time of AFI onset, description of the adverse event, what happened first, what happened next, what happened third, what what is the management given, what is the outcome of AFI, and whether it is fulfilling the criteria to become a serious event. Then coming to the reporter, the name of the first reporter of AFI, and the institution or location or the department where the reporter is working. And for further communication, the email address and the telephone numbers. And if you want to give any other comments, that also can be given. So using these core variables, the Government of India has released the case reporting forms. So it has multiple sections. It is a 2-page document. I have given only the screenshot of the first page. Again, you can see there is a unique identification number, etc. Then there is a reporter or notifier details, the patient details, the vaccine details, and behind we are having the details of the event, seriousness criteria, the signature of the reporter, all that things are coming in the second sheet. So whenever the ground level staff, whenever they see an adverse event, they have to make the case report form, this form, and need to submit it to the agency, to the district immunization office. And this is expected, this report is expected within 24 hours of onset of serious adverse events. Then I told you there is a field worker in the district immunization office. They visit the different places where they received the vaccination or the area where they live, and they will make the preliminary case investigation form and will be submitted to the district immunization office. And then there will be a final case investigation form. This will be done at last once we have all the lab investigations and the details, everything is available. So generally, when they are making the preliminary investigation form, we would have collected the details from the site, or if required, the multi-dose vials which will be sent for investigation, all that. Then once all these investigations and details are available, the final case investigation form will be collected. And these document will be subjected for the causality assessment. So this is how it goes in the government set. So coming to the pharmacovigilance program of India. So we are supposed to report the adverse events in the suspected adverse reaction reporting form, which is version number 1.4, which also has almost similar or all similar points. We have the patient information, the event information, suspected vaccine information, reporter details, etc. And this place is basically for the AMCs to fill. Then if there is a serious adverse event, there is another form and which need to be filled. And you can see here, when it comes down to the form, we can even see what is the date and time or date the event is reported, a serious event is reported to the district immunization office, state immunization office, when did we submit it to PVPI, all these information also need to be present. So this information help the national agencies, the PVPI or immunization technical support unit, to identify the repeated events. So we might have reported once to the district immunization office as well as to the PVPI. So for this kind of purpose, we need to prepare this and need to be submitted to the PVPI. Then if there is a possibility, there are multiple vaccines are involved. So then you cannot say that 6 weeks vaccination. So all the vaccine which is given need to be separately evaluated and need to be assessed to identify which would be the possible for involvement of the adverse event. Or if there is a multiple AFIs present, patient is complaining of fever, patient is complaining of gas, loose stools, etc. There are 2-3 events. Then we need to make a separate report for each of the adverse event. So if there is a fever, loose stools, and headache or 3 different events, then you have to make 3 different events. Okay, so because while doing the causality assessment, we look for the event and the vaccine. So multiple event at once we cannot analyze. So it is separate event need to be reported. And in case of AFI cluster is a similar event occurring in a group of population. So you cannot give a single report of a cluster. Each patient in the cluster should be separately reported and need to be evaluated. So if you look at the AFI flow of information, which I already said, if there is a possibility of minor events, severe events, and serious events. So if there is a serious event, which need to be reported immediately within 24 hours. If there is a serious adverse event, a severe adverse event, it can go. Severity is the intensity of the reaction. So it did not reach to the seriousness. It did not get hospitalized, did not die, nothing happened, but it is the intensity was too high. Then that can be reported in the weekly report from the healthcare worker. Then all these events, including minor, serious, severe, all these events need to go in the monthly report to the state immunization office. And if you look at the serious adverse events, which we are reporting to the district immunization office, so we are reporting within 24 hours. Then the district decision on investigation and they will be informing the state within 48 hours. Then the preliminary investigation form, that is a preliminary investigation form, will be filled or completed within 10 days. And the final investigation will be completed within 70 days of the development of the event, and the causality assessment will happen by 100 days. So within 100 days, the causality assessment will be completed, and that will be made available for the public access. So coming to the causality, so there is a causality assessment criteria or a classification, which is released by the WHO. So especially that particular criteria is used for the post-marketing surveillance. So we can, as I already said, there are different types and causality. So there are 3 different categories. We can divide after doing the causality assessment. There is a long process. There is a causality assessment question. Then there is a causality assessment checklist. Then there is an algorithm. It is a long process. So we need all the information to fill in. I'm not going to explain those in detail. But after performing this causality assessment, we are categorizing the adverse event into 3 categories basically. One is consistent causal association to immunization. Second is indeterminate, and third is inconsistent. So under this consistent causal association, the 4 types of event will come under this: the product related reaction, quality defect related reaction, error related reaction, and anxiety related reaction. So all 4 will be related to the immunization. Then coming to the next category, indeterminate. Again, we have 2 types under that. B1, where the temporal relationship is consistent, but there is insufficient definitive evidence for vaccine causing the event. It may be a new vaccine linked event, which was not identified before or not present in the published literature. So when the vaccine is newly introduced, we will generally look into the fact sheet released by the manufacturer for identification of the adverse events reported during clinical trial. So if it is not reported before, then we can fix into this B1 category. So this B1 can be a potential signal. So which is not identified before, it may be a new event. Coming to B2, there is a qualifying factors are leading to a conflicting trends of consistency and inconsistency with the causal. We are not sure. So we are 50%. We are thinking that it is because of vaccine. 50, we have different things. So we are very unsure of this particular event. So these kind of events need to be further investigated to put under any of these categories. And if the event is nowhere related to the vaccination, that will be considered as a inconsistent causal association, or the type is coincidental event. And if so, these 3 categories is possible when we are having an adequate information on the reported event. And if you are having an inadequate information, which will be classified as unclassifiable and generally will be sent back to the reporter for more information. So if you look at the data sources to monitor the vaccine safety for the regulatory agency, from where they get the sources, it is from the Universal Immunization Program, or from the PVPI, or from CDSCO. So in the National Universal Immunization Program, through the AEFI reporting, it is either monthly report, weekly report, or a direct reporting to AEFI, or IAP, it's a program system. So there is a online system for reporting of ADR. So through this way, the UIP will get the safety data. Then pharmacovigilance, there is through the ADR monitoring centers. So immediate sharing of serious adverse events from the ADR monitoring center to the state immunization programs. Then there is a monthly sharing of the non-serious and other adverse events to PVPI. and to Central Drug Standard Control Organization, CDSCO. So most of the time, the MH, marketing authorization holders, they will be monitoring and reporting the adverse events. So this reporting happens during the immediately after the introduction of the vaccine. Or there will be coordinated review reported to the AFI with the pharmacovigilance stakeholders. So these are the different data sources for us to get the safety reports. So it is not globally we can do at once the monitoring. Every country need to have a separate AFI monitoring and reporting program, because each country will have a different immunization schedules, different types of vaccines or different vaccines. There may be possibility of lot to lot variations or batch related issues, cold chain problems. There is a possibility of errors and different background rate for the possible adverse events, coincidental events, genetical difference. So because of all these things, each country globalize the safety data. Each country need to separately monitor and report the adverse event and come to a conclusion. And communication is very important in AFI. It is basically for transparency and accountability of the adverse events. And the data, whatever we get from the adverse event reporting, that can be converted as a knowledge and can be used for the better patient care. So when you are having a sufficient information and if you are communicating effectively to the public, then we can bring the public trust in vaccinations, which lead to a better decision making for the healthcare professional as well as for the parents or the beneficiaries. So when you are having a better decision, good trust, the coverage will be improved. And when we are having a large data, the rumors regarding the adverse events can also be decreased and a better coverage, and we can reach to a herd immunity. And to conclude, the vaccine pharmacovigilance is not blame gaming. It is to take up the corrective actions to prevent the adverse in preventive action to we do not want our community to get damaged due to the fake news or news about the adverse event. And the data, whatever we generate, can be used for training and for a better communication. And we can prevent the vaccine preventable disease outbreaks or can control the VPD outbreaks if you are having a sufficient data. And obviously, most importantly, the public confidence in vaccines and public trust. Thereby, we can decrease the vaccine hesitancy. So with this, I conclude my talk. Thank you very much for patient listening.
Thank you very much, Junie. I ask the participants if they have any questions related to this particular topic, they can post it in the question answer chat box, and we will take it up in the end of the session. Now we will move, thank you very much, Julie, and we will take your questions in the end of the session.
Sure, ma'am. Thank you.
We will move on to our next speaker. We have our next speaker is Dr. Meera Desai. She is a professor and head Department of Pharmacology, Within Medical College and Research Center, Nagar. Ma'am is a clinical pharmacologist with more than 30 years of experience in medical education, clinical research, ethics, as well as drug safety monitoring and medical writing. She is a GCP expert. She trains and also has experience in the inspection of CROs and especially people involved in clinical trials. She is a subject expert by CDSCO, member expert committee to assess FPCs, chairperson and member of the assembly for phase 1-2 clinical trials of drugs, vaccines, as well as safety committee. Faculty for Skill Development Program and Signal Review Panel, PVPI, National Faculty for WHO Training and Trainers and Coordinators of DRTB and ART Centers. She has received the CDRI Oration Award for Outstanding Contribution in the year 2017 for her publication and research. And she possesses more than 70 publications published in both national and international journals. With this short introduction, may I request Meera ma'am, and I invite you to please present your talk.
Thank you, madam, for those kind words. And can I share my screen now?
Yes, yes, ma'am. Yes.
Can you see the slides? Hello?
Yes, ma'am.
Yeah,
Yes.
ma'am, we can see the slides.
Okay, I'll make it on slideshow.
Yeah, yeah.
Is it fine? Is it okay?
It's not yet done, ma'am.
Oh, not yet done. Uh-huh. Is it fine?
Yeah, yeah, ma'am. Now it's fine, ma'am. Thank
Okay.
you so much.
Yeah. So very good morning to all of you. And it's my pleasure and privilege to be part of this vaccine safety webinar. And I thank Dr. Ramesh, Juni, and the organizing team PVPI for this invitation. And for the next half an hour, we'll be mainly talking about the clinical research and development of vaccines. And this is the outline of my presentation, the overview of clinical research and development process for the vaccines. What is the difference between the vaccine and the drug trials, and the recent experience and approach for COVID vaccine, and what are the regulatory perspective for the vaccine research and development. The vaccines are complex biological products which are given to healthy people, and that's why safety is paramount. While the clinical research and development is a complex, highly technical, expensive, time-consuming, and resource-intensive process. And the key question the clinical research and development should answer is the vaccine safe? Is it effective? What is the dose and the schedule? And this is a slide showing the overview of research and development process. A potential vaccine candidate undergoes elaborate research and development process. World over, the regulatory agencies have divided this research and development process into preclinical and the clinical phases. The clinical phases includes phase 1, phase 2, phase 3, and this happens sequentially. Once the phase 1 is over, then only the vaccine can go into the next phase of development. And after phase 3, the regulatory permission is obtained by the manufacturer and the marketing authorization holder, and then the phase 4 starts. Now, the guiding principle for the vaccine research and development remains the same for all vaccines. However, there can be difference as per the target population, as per the type of vaccine, as per the disease epidemiology, what is the burden in the society, and even whether the pre-existing vaccines are already available or not. Moreover, the transition from phase 1 to 2 and then 3, it has to happen after one another as per the data generated in each phase, reviewed and approved by the regulatory bodies. Then only you can proceed further. Now, with this, let's try to find out what happens in each phase of clinical research. The strategies and objectives of phase 1 trial is to evaluate the vaccine safety, the reactogenicity, which means the inflammatory reaction, which is expected, the local reaction, especially at the site of administration, which can be pain, redness, erythema, and even systemic inflammatory reaction manifested in the form of fever, body ache, headache, and the immunogenicity, the immune response, the development of the antibody titer to the vaccine. And this is done in a small group of 20 to 80 healthy, uninfected adults. So this happens in phase 1. Secondly, in phase 1, we can also assess a safe dose range. For example, the adults, they are exposed to dose 1 level, and then gradually the dose is increased to the same individuals, and that's how the dose range is identified, which is giving the optimal immune response. And this helps in identifying the dosage schedule and the mode of administration. Thirdly, in phase 1, it is first tested on healthy adults who are immunocompetent, who are least likely to be infected. And then, based on the study objective, as well as the proposed use of the vaccine later on, the age of the test subject is gradually stepped down. What I'm trying to say is, initially it is tested on the adults, healthy adults, followed by adolescents, children, and then infants. Further, usually phase 1 vaccine trials are conducted in the tertiary care centers or in the CRO, Clinical Research Organization, having a complete setup for phase 1 studies, which is approved by the regulatory authorities. And initially, the volunteers are very closely monitored because there is always a safety concern. Vaccine, most of them are administered parenterally, and they are likely to cause allergic manifestation or hypersensitivity. And that is why initially the volunteers are closely monitored for safety assessment, adverse event, tolerability, reactogenicity as per the toxicity grading scale and case definitions. Further, the volunteers who develop the immune response above the certain threshold level are also assessed. If I try to explain this, the baseline antibody titer is already done, and then following the dose administration, the vaccine administration, what is the increase in the antibody titer as per that, which is the dose which causes the optimal level of antibody titer, because it never happens that you increase the dose. That does not mean the antibodies titer will increase, because the body has a limited capacity to develop the antibody response. Moreover, age, above the age of 55, again, there is a limited capacity of the body to develop antibody. So which is the dose which causes an optimal antibody response, that is also identified. And one caution about the phase 1 trial, especially for the live attenuated vaccine, the volunteers should be closely monitored because there is always a risk of developing infection into the volunteers. Moreover, if there is a shedding of the virus in the body secretions, even the close associates of the volunteers, the family members, they should also be monitored, or a history should be taken for developing the infection. And if the results are promising from phase 1, it moves ahead after submission of the data, getting it approved by the regulatory body, the phase 2 trial starts. The objective of phase 2 study is to evaluate again the vaccine's safety, immunogenicity, the proposed dose, the schedule of immunization, and the method of delivery in more number of individuals. And this can be several hundreds. In phase 1, it is few, less than 100. Here, it is more than 100. And phase 2 studies are usually conducted in the community so that you can also have an additional data and the study population can be adults, adolescents, depends upon the objective of the study. Phase 2 studies are randomized, well-controlled, and placebo group. And then the phase 3 study starts. And in this phase, it is more sort of a real world situation. It is mainly to confirm the vaccine safety, statistically robust efficacy in a significantly large group of people in prevention of infection. What I'm trying to say is whatever is observed in phase 1 and 2, before the vaccine is approved and available in the market, we need to confirm that yes, it is definitely safe and it is definitely having an immunogenic effect or immune response. And that is why the study is conducted in minimum of the 3 to 10,000 of the people, including those with multiple comorbidities who might be taking multiple drugs, as well as those who are also at the risk of infection, let's say elderly or having some comorbidities, and that will reflect a sort of a very close to a real life situation. Phase 3 trials are usually multi-country, multicentric, randomized, double-blind, placebo-controlled or active controlled, and active monitoring, follow-up is very, very essential to assess the safety and tolerability. The concomitant administration of other vaccines as well as other drugs is also allowed. So what is the impact of the other factors, the age, the administration of other drugs, administration of other vaccine, or even the presence of other clinical conditions, comorbidities, all these factors are also studied. Subsequent to the successful phase 3 trial, a dossier is submitted to the regulatory authorities by the sponsor, and the central licensing authority, they review and approve the vaccine. And obviously, there will be a good number of discussions. There may be some recommendations for additional data, or if the data is adequate and very robust, it can be approved by the central regulatory authority, and then it becomes the responsibility of the central licensing authority to monitor the production of vaccine, inspect the facility where the manufacturing is going on, and the whole manufacturer's testing process. That becomes again the responsibility of the regulatory authority. And then once the vaccine is approved, available in the market, used for the general population, it comes under the domain of vaccine manufacturer and marketing authorization holder, and the phase 4 or post-marketing study starts. And this becomes a pharmacovigilance activity for a continuous assessment of the vaccine safety. The various methods which can be used are passive surveillance, active surveillance. There can be adverse event of special interest. And this happened especially with the rotavirus. There were few reports, and that's why there was an active surveillance to find out whether this vaccine, there is a affinity relationship. If 1 or 2 cases are reported, then this becomes an adverse event of special interest, and the special monitoring is done. Or adverse event in special population, high-risk patients having multiple comorbidities or something like that. And then ultimately, when the vaccine is available to lakhs and millions of people across the country and the world, it really helps in determining the herd immunity. What is the protection offered, which we tell it as an effectiveness, the protection offered by the vaccine. It is not efficacy, but effectiveness in the general population. So with this background, the question comes is there any difference between the clinical research and development of vaccine and the medicines or the drug trials? Because both are medical products, both can cause adverse events, both have the potential for interaction with the disease, drugs, and the vaccine, both have to prove their efficacy and safety, both have to comply with the standards of safety, efficacy, and the quality. So if you look at the general guiding principles, it is almost same. The process, the path remains the same. However, there are certain differences. The first and foremost thing is the target population. And as I've already mentioned, usually vaccines are given to the healthy individuals. They can be healthy adults, healthy adolescents, healthy infants, healthy children. And that is why they need to have a high safety margin. Otherwise, the vaccine will go out of the vaccination program. Secondly, you always need to have a step-down approach. First, always test on the adults. And then as per the proposed use of the vaccine, the age of the test population is gradually stepped down. That is adolescent, children, and infant. Further, especially under the universal immunization program, and if the vaccine is to be tested on the children and infants, these test population, the study population, will also receive other vaccines under this national program. And this needs to be considered in your research and development in your protocol, whether it should be allowed. Obviously, it has to be allowed. Otherwise, it will be unethical. The ethics committee will not approve it. So if it is allowed, whether it should be administered during the same time when the baby comes for vaccination in the pediatric OPD, or there has to be some distance, some time interval between the two vaccinations. Further, whether it has to be administered at the same site along with other vaccination, or it has to be administered on some other arm, like the test vaccination is to be administered on left arm, and the routine vaccination under the national program has to be administered on the right arm. Otherwise, this can result into interpretation, because here our objective is to assess the local reactogenicity. So whether it was your test product or the routine vaccination that resulted into local reaction. And this has to be trained to the local clinical site staff members. I have seen that they make the error, and this is what Junie was talking, there can be administration error. Now, how do you interpret if both the vaccines are given at the same site, at the same time, whether it is your test product or it is whether the routine product? And that is why it requires extensive training and monitoring of the clinical trial site. Further, the live attenuated vaccine, as I've already shared, there is a risk of infection, and that requires a close and long-term follow-up. And all of them being healthy, you need to have a strong subject tracking capabilities. There has to be a system in your research and development that how are you going to trace the subject so as to collect the data for safety. Otherwise, a good number of your subject will be lost to follow. And this can be a logistic or operational challenge at the clinical trial site. However, it has to be a part of the research and development process. Otherwise, a good number of your volunteers will be lost to follow-up. Further, the vaccine stores require refrigeration, some minus degrees, minus 20 to minus 70. You need to maintain the cold chain to remain them stable. And that this type of, again, operational challenges, the logistic issues has to be incorporated. Otherwise, the efficacy will be lost. And unlike drugs, usually when a drug is introduced in the market, they undergo bioequivalence and bioavailability study, pharmacokinetic studies. For vaccine, there is no pharmacokinetics, but we need to determine the kinetics of immune response. And what is this immune response? That is a protection offered, the immunity offered, the immunogenicity, the antibody titer. And obviously the body will take its own time to develop immunity. It will not be detected immediately. And again, this is one factor. You need to follow up the patients for a long time. Moreover, even the immunity, the antibody titer, how early it develops. For example, at dose 1 level, you start with, let's say, 25 microgram. At what point of time you should assess the immune level? How early it detects? How long it will sustain? Which will help us in determining the dosage schedule, the primary dose, the secondary dose, or a booster dose. For example, for a pediatric patients, for infants, it is usually 3+0. That means 3 doses at the age of 6, 10 and 6, 9 and 10, or 6 and 10, and then a booster dose at 18 months. So all these factors will be determined by the research and development, and we need to assess, and this has to be incorporated in your protocol. Moreover, as Junie already described, vaccines, they have multiple ingredients: antigen, suspending fluid, preservative, stabilizer, adjuvants, and each of them have a bearing on the safety of vaccine. If these adjuvants or all these ingredients are already being used, not much to worry. But if the adjuvant, which is used to boost the response, to accelerate the immune response, and many times the adjuvants are added just before administration of the vaccine. And that is why, again, this is an area which requires training. You need to have a trained pharmacist, a trained nursing staff for mixing preparation of the vaccine just before administration. Otherwise, this can have a problem in the efficacy as well as safety. And obviously, everything ultimately revolves on the safety of the vaccine. The assessment for the safety should be robust and standardized. In phase 3, there will be multi-centric. Each investigator have a different perspective to notify, to report whether the fever, whether it was mild, moderate, and severe. If one side reports severe, the other side reports moderate. This will really interfere in the assessment. And that is why there has to be standardization. And this has to be incorporated in the research and development program. There has to be uniform criteria for categorizing adverse event to compare the safety data among the groups within the same study, as well as to compare between the different studies. And there also has to be inclusion of case definition for many adverse events following immunization. For example, diarrhea. Diarrhea, how many diarrhea? Whether it is associated with fever, constitutional disturbances. So whether, what type of grading will you give? This has to be standardized. It has to be incorporated. Grade 1, 2, 3, 4, mild, moderate, severe, and life-threatening. What is the criteria? In which circumstances you will label it as mild? In which circumstances it can be labeled as moderate? And in which factors, especially if it is fever, diarrhea, abdominal pain, number of diarrhea, consistency of the stool, whether it happened within 24 hours or within 2 to 3 days, all these factors has to be considered and incorporated in the tabular form. That will help the investigator to report and assess whether it was the adverse event was mild, moderate, and severe. And this will not only that, it will also help the program managers, the sponsors, to find out whether the adverse event is mild, moderate, severe, which needs to be considered immediately, or this can happen, or it can be, just needs to be reporting. All treatment emergent adverse event, whether they are solicited, unsolicited, local or systemic, happening in test group or the reference product, the type of adverse event, as I already mentioned, mild, moderate, severe, or serious, or life-threatening, and the relationship to the product. And this is very well described by Junie, whether the relationship is to the vaccine product, the investigational product, whether it is related to the vaccine product or some other quality issues, immunization anxiety related reaction. Again, this is especially after the age of 5. Usually under the age of 5, the baby will not have any anxiety for the reaction. This is usually reported after 5 years of the age. The temporal relationship, inconsistent causal association to immunization, or it is simply a coincident, and this can happen, especially in the pediatric vaccine program. They are likely to become sick. They may suffer from upper respiratory tract infection. So it is just a coincident, and case without adequate information for causality assessment, that is unclassified. And this has to be, this detailed information has to be reported in the clinical research and development. And all adverse events should be coded as per MIDRA coding system. So with this, let's try to find out the recent experience and approach for the corona vaccine. I'm sure all my participants will agree with me that last 3 years has been a nightmare for all of us. And the only thing which saved us, today we can say it was corona vaccine. So what has really gone into this? I've already shared with you the overview of the research and development. So what has been the experience with the corona vaccine research and their availability? If you look at this slide, it shows a very stepwise approach. It is happening in the sequence. Once phase 1 is done, it goes to phase 3 and then phase 4 and then registration. And this is the slide showing the conventional pathway for vaccine development, which is somewhere around 10 to 12 years. That is a minimum time period. Interestingly, with the corona vaccine, the first case of corona was reported somewhere in some corner of the world was in December, end of December 2019. While the first case in India was reported on 27th January 2020. And the first vaccine available for corona was in January 2021. I still remember the date, which was announced by our Prime Minister, that from 16th of January 2021, the vaccination was started to the high risk, that is mostly the healthcare professional. So if we look at the time period, it was only 1 year. And the speed at which this corona vaccine has undergone the research and development and made available to the people across the world, it is remarkable. This has become a history. This has never happened in the history of scientific community that a vaccine can be made available within a year. Although I would say that the knowledge of the spike protein of the virus is responsible for the pathogenesis of corona. And if we can develop the antibody against this spike protein, it will be really helpful. The knowledge was already available, but the research was not there, and there was not a single vaccine available at that time for corona. How this has happened? This has become history, I would say. It is an unprecedented collaborative effort among the local and the global stakeholders, the government, the regulatory bodies, the industry, academia. They all worked relentlessly on a common platform with the commitment to come out with a vaccine. And that needs really appreciation. Moreover, if we look at our local situation in India, the regulatory system in India, the regulations, it allows the relaxation and deferment of data, especially in emergency situation for the emergency use and licensing for unmet medical needs. Where there are no drug treatment, lakhs and millions of people were dying in India and across the world, and there were no drugs available, no vaccines available, and COVID-19 justified that this was a situation we can use this regulation, which is available in our new drugs and clinical trial rules of 2019. The regulatory system in India has a provision of accelerated approval process that matched the current pandemic. So it was a proactive approach by the stakeholders. The different steps of the vaccine research and development, it was continuous, parallel, rather than sequential. This is a very important difference. I have shown you in my previous slides that it goes from one phase to another. It happens from one step to another step in a sequence. However, with the corona vaccine, it was parallel. There were multiple things were going on, like clinical evaluation, different phases of clinical research and development, safety evaluation, the production, planning, distribution, everything was going parallel, and that has really helped. Secondly, the use of innovative adaptive designs, the seamless immunobridging studies to demonstrate the equivalent immunogenicity in a demographically diverse healthy participants. If I try to explain this, what has happened practically? Normally, what happens after the phase 2 only, the phase 3 starts. But here, there was a bridging studies. There was an immunobridging and seamless. There was a combined phases of the trials were allowed. And this was allowed based on the regulatory provision in India, as well as I would say other regulatories. There is this has happened that it was the seamless immunobridging studies. The study which was going on, let's say in Netherlands, the same phase with the more number of population was also going on in India. So at that point of time, instead of having 80 healthy subjects, we were having more than 100. We were having thousands. In phase 3, the minimum requirement is 3,000. But with corona vaccine, I have seen and I have read somewhere that the minimum participants, volunteers who were exposed in phase 3 trial was somewhere around more than 20,000 at a time, because the diversity, demographically different countries. Parallelly, the study was going on in USA, in European countries, as well as Asian countries. So at a time, the data was generated, and that's how we were able to evaluate the immunogenicity as well as safety. And this is the slide showing the intelligent and innovative use of adaptive designs. This is a design of how the study flows. As for example, this is the bridging study. The number of subjects involved recruited are 100 only. They are exposed to dose 1. And these subjects are observed for a particular time point. This has to be specified in the protocol. And at the end of that point, the data collected is analyzed, reviewed by the Data Safety Monitoring Board, that is submitted to the regulatory authorities, and recommendations made by DSMB. If it is approved, the study progress ahead with the same dose on more number of subjects. And this is actually going on. They are observed again, you can see, and this will be randomized and blind trial, and they will be exposed to dose 1. On the other hand, the 100 subjects at the end of that time point, they were exposed to dose 2. And the study progressed further. The safety data was collected, again analyzed at a particular time point specified in the protocol. The DSMB was shared with the safety data assessment, and that is again reviewed. And at that point, again, this more number of subjects, they were started on those 2. And that is how it went. So what I'm trying to convey here is these adaptive design. This allowed us to use parallel, combine different phases, and we could recruit more number of such subjects at a different dose level. And that's how the data collected was increased, and the time period was compressed. The duration for the vaccine trial for one particular phase, which is somewhere around 6 to 7 years, was compressed to, let's say, 5 to 6 months. And that is the beauty. And that is the reason why this vaccine is now made available to all of us. There was, I would say, I would also like to share that there was a public apprehension. And I think in many countries, people were reluctant to take corona vaccine. What was the reason? The reason is that because normally the conventional path, which takes 10 to 12 years, how could they compress 1 year? That means they have compromised the scientific evaluation, and there can be the regulatory standards might have been down. And this has been documented, you see. And this was an apprehension among many people across the world that we don't want to take vaccine. But no, there was no compromise, I would say. It is because of the combined extraordinary efforts of all the stakeholders across the world, as well as an intelligent use of the adaptive clinical trial designs. So with this, let's try to find out what are the regulatory requirements and how do the regulators look at this vaccine studies. Yes, As I said, there is no compromise for the vaccine trial, whether it was a corona vaccine or any other vaccine which is under the clinical trial. All vaccines have to follow the guidelines as per the Drug and Cosmetic Act 1945, as well as new drugs and clinical trial rules of 2019. Moreover, if it is a recombinant product, the vaccine should also comply the requirements and guidelines which are prescribed by Department of Biotechnology. The regulators, whenever any data which is submitted, they look at the data. From this point, the data has to be robust. It has to be adequate to demonstrate vaccine efficacy. That is an acceptable correlate of protection. If it gives 100% protection, that's very good prevention of the disease, totally. However, it is difficult. But at least if it offers 50 to 60% of the protection, and that will depend upon the sample size, the data which will be submitted to the regulators, they will look into the sample size, the power of the study, and the antibody titers. What is a significant level from the baseline? What is the antibody titer which has developed in a particular time period? And obviously the vaccine safety. What are the potential risks? Because vaccines, they are likely to produce local reaction. Covishield or Covaxin, there will be pain, there will be some fever. That is normal. And that is acceptable, but whether it can cause something serious, they will look at it, which is it is a risk to the people, general population. So these are two important factors, but they also look at the study power, the sample size, the robust data, and the type of assessment and the recommendations by DSMB members. Further, any new vaccine which is discovered and developed in India should undergo phase 1 study. That is definitely as per the drug. You have to start from phase 1. However, if a vaccine is developed and undergone clinical trial outside India, the data generated will be considered for evaluation. The local domestic trial can be waived. However, it will be the obligation on the part of the marketing authorization holder to do post-marketing studies. That is, for the drugs as well as vaccine, the regulation remains the same. The permission to conduct clinical trials should be phase-wise, from one phase to another phase. The corona vaccine, even it was an emergency situation, each and every phase has undergone the DSMB recommendation, approval, and followed by the regulatory approval. It has to be, after that only, as per the data generated in earlier phase. The interim data review and the recommendation by the Data Safety Monitoring Board is very, very essential. And as per the US FDA regulation, the study population before the study is giving a marketing approval should be minimum 3,000 individuals for pre-licensure assessment. However, I would like to say most of the studies, the vaccine will be evaluated. Not 3,000 is the cutoff, but it will be more. And as I said, for corona vaccine, it was assessed in more than 15 to 20,000 of the individual. So we can say that the vaccine development has moved at a record speed through strong public and private partnership. And currently, we have, I think the vaccines which are already in the market for general people is more than 7 to 8. And currently, there are more than 200 vaccine trials are undergoing research and development and in trials. Hence, to conclude, the clinical research, development, and the regulatory requirements for vaccines, they are almost same as drugs. However, the difference is the target population, healthy subjects, the efficacy assessment, that is immunogenicity, the safety data assessment using uniform criteria for categorizing adverse event and case definitions, they are different. Further, the operational challenges like cold chain maintenance, strong subject tracking system, risk of contracting infection in case of the live attenuated vaccines, this require long-term follow-up. The recent COVID-19 vaccine has proved it has become a game changer that the use of adaptive, seamless immunobridging study designs are more efficient with reduced timeline for the development and bringing an effective vaccine for the people. And it has been, I would say, it has been a boon to all of us. Thank you very much.
Thank you, ma'am. It was a very extensive talk with your full experience, which will be a very good guidelines for everybody, the participants who are attending today. Thank you so much, ma'am. We will take up your questions in the end of the session.
Sure.
Is it okay? Yeah.
Okay.
Thank you.
Yeah.
So we will move on with our next speaker of the day. We have Dr. Madhavaram D. Sir is placed at Department of Regulation and Pre-Qualification Assistive Medicine and Health Products, World Health Organization, Geneva, Switzerland. Dr. Madhavaram Balakrishnan is a medical epidemiologist, an international scientist, and an expert in vaccine safety. He has pioneered the WHO AEFI causality assessment methodology and the development of several tools, training, and software for vaccine safety. Sir has been actively involved in the development of global guidelines and manual for COVID-19 vaccine safety, including investigations, data management, and causality assessment. He is also a member of WHO editorial board for immunization, vaccine, and biologicals, and contributes to the development and update of WHO position papers on vaccine. This is just a short introduction. Sir, I welcome you for this, and I request you to deliver your talks.
Thank you very much. Can you hear me?
Yes, sir, we can.
Wonderful. 1 minute. Let me just see. Can you see my full screen? Is it in the full screen mode?
Yeah, it's in the full screen, sir.
Excellent, excellent. So first of all, good morning. It's actually for me, it is about 8:30 here in Geneva. And first of all, let me just thank you very much, especially the organizers, for giving me an opportunity to share with you some very, very useful and interesting information on vaccine safety, particularly 1 minute. I think it's a wrong presentation. Just let me get the right presentation on board. All right, 1 minute, let me. Yes, this is the presentation, because usually my opening screen is the same one. Right. So basically, thank you very much, and it has been very, very interesting listening to all of you. I want to especially thank Dr. Ravi, who has also asked me to give this particular talk, and I also wanted to thank Juni Sebastian, who has organized this session. Plus, on top of it, I was very, very impressed with the first presentation which she made, which has made my work very easy to say the least. It has become really very comfortable for me to make this presentation. Also, I was very impressed with the presentation which was made just now by Dr. Meera Desai. Very impressive, covered quite a lot of areas, and very comprehensive as well. So let me, without beating around the bush, let's get down to bar stats. No, a couple of, when this conversation came up, people were, in fact, the organizers asked me to talk about causality assessment for AEFIs. Now, one thing which I realized was, we cannot really talk about causality assessment in 30 minutes and, do justice to that particular job. It really needs, we have something like in WHO, a 4.5-day training program exclusively for that. We have conducted it in India, I think around 3-4 times. Instead of this, I was just trying to, what I'm going to be talking about is very interesting, and it is going to follow what Juni Sebastian presented. In fact, I'm going to use 1 or 2 of her slides as a starting point to make my presentation. And let me start off with this, okay? So we are going to look at it from a very, very practical perspective. We are going to ask this question: what happens to AEFI reports which I submit to the government? People like you, people like me, we fill up this reporting form and then we send it to the next level. Now, what really happens to it? How is this decisions taken? That is one question which comes to people. The other thing is, you also are going to ask me questions. We heard recently that there is a connection between the viral vector vaccines as well as this thrombosis thrombocytopenia syndrome, TTS. We also heard about this pericarditis and myocarditis connected to the mRNA vaccines. The question which comes up is, how is this actually decisions made? So we are going to be focusing on that. So let me just start off with the basics. So in the next 30 minutes, we are going to be looking at 4 things. We are going to be touching on the basic concepts, and I'm going to touch on a few slides, a couple of slides which Juni Sebastian presented. Then I'm going to talk to you about the actual practical aspects of data collection and processing from the field. From the field, it means like when you fill up the national AEFI reporting form, what really happens? And when it reaches the national level, what happens? And when India sends it to the global level, what happens? So this is what we're going to be talking on. And how is this data used and decisions made? And also one more important thing is, if you're working now, you are maybe working as a state level head or a institution level head or even a national level head. Now, what should we do exactly? So that's what we're going to be touching on. So let us first of all touch off with the basics. Okay, and let me start off with this slide, which was presented by Junie just now, because we need to understand the fundamentals and the basics. So the thing is, this is important to understand. That is, an AEFI is an untoward medical occurrence which does not necessarily have a causal relationship with the usage of the vaccine. See, the most important words here is does not necessarily have a causal relationship. What does it mean? It means that the vaccine is innocent until proved guilty. That is, you cannot attribute an adverse event to the vaccine unless you establish a causal relationship. And as Junie mentioned right now, the event can be a symptom. Like when you look at this event, you need to define it as a symptom. Symptom is what you complain, what a patient complains. It could be itching, it could be sneezing, it could be something like that. It could be a sign, like a hepatomegaly, or it could be a sign like, you auscultate and you can find lung signs or something like that. So it could be a lab finding, like what you see on the picture is actually thrombocytopenia, but it could be something like, let us say, ECG changes. It could be an MRI changes. So these are the kind of things which you can find out. Or it could be a disease like purpura or meningitis, or VAP, vaccine-associated paralytic polio, something like that. It could be any of those things. So this is what it is. Now, this is the slide which was also in a different way presented by Junie, and I'm so glad that she presented it so beautifully. But let me just go through that once again. Now, what happens is the story begins with this orange arrow where the child gets, or it's not the child now. With COVID, it has become everybody. Now, the person gets vaccinated, and then there is a stage where the event is detected. Then the second stage is the stage where the event is brought to the notice of the health system. And when it is brought to the notice of the health system, it is seen either by a health worker or by a nurse or by a doctor. It could, in some countries, even that there is a patient reporting mechanism. And what really happens is there is an AEFI reporting form. If you remember Junie's presentation, she did show the form to us. And she also mentioned something like it is a 2-page form, if you remember that very clearly. Now, what really happens is in WHO, the form what you see on the screen is the one which is actually used. Okay. Now, this particular screen, it has got 25 core variables. Now, if you notice, Junie also did mention something about the 25 core variables. Now, remember something, when you collect this piece of information, you're collecting minimum information. You're not collecting all the information. The idea behind that is, during reporting, when the report from this particular paper goes to your supervisor, the case is categorized into 2 categories. Junie also mentioned this. It is divided into 2 parts. One is it serious? Is it non-serious? Which are the serious cases? Serious cases are only 5 things. It can be a death, hospitalization, disability, congenital anomaly, or any other medically significant event. Now, only if it is a serious case is it investigated. This is very important, okay? So you may report about 1,000 adverse events, and maybe only about 5 of them might be serious. It might fit into that category, and only that is investigated. Now, when you look at this investigation, you should collect all the details. Whereas when you look at the AEFI reporting form, you are only collecting minimum information. And basically the idea is you want to know the time, the place, and the person. Who are affected, where are they affected, which is the vaccines. And you remember again, Junie did present that core variables, and she beautifully explained that in 2 slides. So that is what we are trying to do during reporting. But during investigation, you should collect all the information which is possible, because investigation is used for causality assessment. Then you are having a stage of data analysis. Now what happens is, from these reports which are collected, this AEFI reporting form, and also from this investigation, at the district level, there is a guy who sits there, not usually the district immunization officer in India, whereas in different parts of the world, you have different people who actually, who will be doing this. Now, then this data analysis, we'll be touching upon this because it's in the next few slides, so I'm not going into too much of details. Now, the next thing is, after this, Among the serious cases which get investigated, they do a causality assessment. And this was also beautifully described by Junie. You remember there was a slide which said, consistent, indeterminate, inconsistent, and unclassifiable. So it was beautifully described. So the idea is this kind of causality assessment cannot be done by an individual. It has to be done by a group of experts. So usually in India and all, you'll be having groups like, in Tamil Nadu, there's an AEFI committee, Kerala AEFI committee, Karnataka, etc. And also there is a national AEFI committee, which is actually coordinated by the ITSU, Immunization Technical Support Unit. So these are the guys who, and also in the Ministry of Health, there is a group which actually works independent of the government, independent of the EPI program, and independent of the regulators. So these are the people who do the causality assessment. Now, let us start off the story. So you need to collect data and generate information for action. So let me go back to this. So remember, you have this AEFI surveillance cycle. I'm bringing it back into the next slide. And then you need to collect the data to generate information. So how is it collected? So there is a source of data. You collect it, you interview the patient, you look at the records, and then what you do is you transcribe it. You fill up that form, what I just showed you. You send it to the next level of hierarchy. And that is where, there is in India, I know for that matter, there is a software, I think it is called the VAMES, Vaccine Adverse Events Information Management System. So what they do is this is filled online. And then once it is filled online, the data enters into a database. And then when it reaches your next level, so it might be entered at the district level, but when it is reaching the capital of the state, the data is, and even in the district, it is reviewed, and then they check for mistakes and inconsistencies, and then it is auto-generates the graphs and tables. And once it is generates it, then what we do is we get line lists which are available, which this line list will be able to again slice and dice the data further, and then the decision is made, And what is the decision? When you mean the decision, what exactly do we mean by that? This is related to the causality assessment classification, which Junie shared with you just now. And then what happens is, when you actually right now, there is an electronic system for that. And then we can generate a PDF file. And then using that one, we explain the logic and decision to the public and stakeholders, and feedback is provided. So this is what it is. Okay, so I have now touched upon the basics. I have now finished the basics about what is an adverse event, how is it reported, and how is the data collected, etc. Now, the next thing which also happens is, we are going to look at how is it collected from the field to the national and global levels. So basically, what we are going to do is, when you look at it at the country level, the idea is we want to make decisions at country level and at the global level. So so when you look at data, there are two types of data which is collected, okay? One is actually the aggregate data, and the other one is the case-based data. Now, what is the meaning of this word aggregate data, and what is the meaning of the word case-based data? Aggregate data means we only know the total number of cases occurring. So for instance, if you look at the case in case of Karnataka must have reported, let us say, 50 AEFI cases in the month of February. And out of these 50 cases, 2 of them were serious. And among the 2 of them which were serious, 1 of them was death, 1 was hospitalized. So this is actually the aggregate data. Whereas when you look at the case-based data, we need to have the individual details, like the patient details, the 25 core variable details will be required for each and every case. So let us look at how aggregate data and case-based data is processed at the country level and at the global level. So this is the same story, and this was also beautifully described by Junie just now, and I'm just going to follow up on what she just mentioned. That is you're going to have this AEFI reporting form, and then it goes into the district, and then from the district, it goes into the state, okay? And then it is, as I said, first it is paper reporting form, which is filled up, then it is filled up, made into electronic system, then it reaches the state level, and this is your state EPI program, and then you might have a state regulator, sometimes state disease surveillance systems. Then it reaches the national level. Now, if you look at it, there is a national database which is available. Like, you have servers, if I remember right, I think in India, it must be located in NCDC. Please correct me if I'm wrong. But this is what happens. So then the data is then shared between the EPI program as well as the regulators. Okay. And then what happens is the regulators, as Dr. Meera just mentioned right now, they also get this information coming in from the manufacturers. And then what happens is then it is approved at the national level. Okay. And once it is approved, the WHO gets the data through something known as the JRF. What is the meaning of the word JRF? It is a joint reporting form. The aggregate data, so the total number of AEFI cases which was reported last year, that particular number is collected with the JRF. And then this is then shared with the WHO India country office. And then it is shared into the regional office. Again, in India, for India, it is in CRO, which is actually located in Delhi. And then there is a WHO uses an electronic system called WISE, WHO Immunization Information System, and then it reaches us at the in WHO headquarters here in Geneva. So this is what happens. So now please note what I'm talking about is aggregate data. Now let us look at the case-based data. The case-based data also, the reporting form usually gets transmitted this way, and then it comes up to here, okay, into the national level. Now, this is where there is some changes which occur. There is some little bit of a difference between aggregate and case-based. Now, you should understand this is very sensitive data. You know, Dr. Meera was mentioning right now, that, they are collecting a lot of details and we cannot keep on sharing this with other countries. There is a lot of confidential data which is being shared. So what they do is there is a data transformation. Now, what is the meaning of the word transformation? The data is actually encrypted. And this particular data, which is encrypted into a format, which is, I don't want to go to details, it's called the E2B format. And then it is shared into the WHO global database. Okay. And this is the WHO program for international drug monitoring. They use the software called VG Base here. Okay. They're using the software here called VG Base. And also in country, they use a VG Flow software, but let's not go into those details. And this is then shared into the WHO headquarters in Geneva, where it goes into relevant committees. Now you can ask me what are the relevant committees. There are two committees. Okay, one is the Global Advisory Committee on Vaccine Safety, and the second committee is actually the SAGE, the Strategic Advisory Group of Experts. Now these committees, which are sitting here only, they make those important decisions. What they do is they collect the data coming from different parts of the world. I cannot, some of the data is confidential, but let me tell you something. One of the huge databases for AEFI actually came from COVID vaccines. We have something like about 3.6 million records of AEFI related to COVID vaccines. And using this particular data, which is actually processed, these committees will then make a decision about, this thrombocytopenia syndrome, pericarditis, myocarditis, and all that stuff. So this is what it has done. Now, there is some challenges, when you look at JRF data, the most important challenge, if you ask me, is what is given on the bottom of the screen. This is very, very, very important. In fact, I have highlighted this in yellow. In many countries, the EPI program and the regulators work separately, and they are not sharing the data among themselves. But for the world, this sharing of information between EPI program and the regulators is very important. So this is, so when you look at it, because, the JRF data, we also collect only aggregate data. It is available only once a year, so it becomes a bit of a problem. And we don't really know the details of those cases in JRF data. Whereas when you look at the case-based data, that is the VG-based data, the quality of data which we get in the global level, in many countries, the quality of data is not very good. Why? Because the reporting forms are not filled up properly. So that is the problem which we face, partially reported forms with errors and all that kind of thing. So we face this particular problem. And, and another problem which we face is, if you look at this 3.6 million records, which I just mentioned, most of those records actually came from United States, Europe, Australia, Japan. We really did not get many cases coming in from Africa, from Middle East, and all that kind of thing. So South America, we don't really get. So these this is a bit of a problem, and then and as I said, it is not shared with relevant stakeholders. Since I have short time, I'm not going into too much. I think I have only another 10 minutes. I'm not going into too much of details with regard to these maps, but I'm just telling you how these maps are just shown. You can see that, from WHO side, we generate these maps on a yearly basis. Okay, once a year, we generate these maps. And then if you see, this kind of tells you what is whatever data we produce, This one, for instance, tells you which countries have got an AEFI reporting system. This is the countries which have got adverse review committee, the National AEFI committee, which is available in different countries. And what is the source of the data? Is it coming jointly from EPI and NRA? Is it coming from EPI? Is it coming from NRA only? So these kind of data is collected. We also have countries which are, the serious AEFI cases which are shared between both the JRF and VG base. And also we also calculate the rate, the per million population, how many AEFI cases have been reported and all. And as you will see, with regard to the JRF reporting, India is doing quite well, if you ask me. And with regard to this VG-based reporting, of course, there's a little bit more work to be done. So this is how we collect the data. I don't want to spend time. I can go on for hours on this. But let us look at it. So how is this data used and decisions made, So let us have a look at that. Now, when you look at this, what we do in WHO headquarters is, when you collect the data, in our, for instance, in the team which I am in, what we do is we actually have a weekly. Earlier, when COVID was very intense, it used to be daily. There used to be a daily briefing in those days. Now it has become weekly. Now the thing is, we collect information from multiple sources. Okay, we collected. This is vaccine-related events which we have. We have even observation systems. All what you feed in Facebook, what you feed in Twitter, everything. We have a software system which actually monitors this. And then we collect this data, we generate autographs, we try to find out in which country is there a peak, is something abnormal which is going on, and this is how we monitor it. Then what we do is the Ministry of Health, we collect and we get reports directly coming in to us from multiple countries. Like in Israel, recently we had some reports, we had reports from Nigeria, and also recently from Guinea, Equatorial Guinea. We also get regional office reports and Uppsala Monitoring Center, the global database which is located in Sweden. We have also developed this platform. This is the platform I was telling you, where we look into the Facebook et al. And then we find out and then the data, we categorize cases, identify cases of interest, and we identify early warning system list. This is then discussed by the GACS Secretariat, which is a group of people who decide if the data has to be shown into a big committee, and then it goes into another committee, which then makes the big decisions. Okay, so this is what happens. So this is how the data is collected, it is processed, it is used. So your reporting form which you fill up, remember, it is going all the way into a global database, and decisions are made based on that. So what are the decisions which were made? In fact, this is a shortened list. If you just look at it, these are some of the committee meetings which we had just in the last 1 year or so. And you see, this is just a very shortened list. In fact, there are much more number of reports which come in. So you look at, you find out myocarditis and pericarditis, blood coagulation events, Guillain-Barré syndrome, influenza-like illness. So many events are actually monitored very carefully, and then decisions are made. So this is it. So now let's go to the last part of my talk, because I know that I have approximately only another 5 more minutes. So what should we do, right? Now, as a person, what exactly should I do? Or if I'm sitting in the national level, if I'm sitting in a province level, or in a state level, or if I'm sitting in a district level, what should I do? For actually remember something, information is no use if you don't share that information. You just collect this AEFI data and then you keep it to yourself and say, what, I've got 50 cases. What is the use? It's absolutely useless unless that data is shared. So let us just start where the data is shared. The data is actually shared here. You need to analyze the data, you need to make your decision, and then the feedback which you collect, that needs to be shared. And for this, what should you do? Whether you're sitting in a state, district, or a province, or even an institution level, make sure you share the data with relevant stakeholders. Because once you share the data, if the state shares the data with the national level, the state can demand information from the national level. So it's very important. Similarly, if India shares the data to the global database, India has the right to ask WHO to say, I want this information, and we are obligated to provide that information to India. So this is very important. It's also important to talk to the stakeholders and determine if the findings are different in other countries and other settings. So what happens is, Dr. Mira's presentation, which she mentioned very clearly, you're getting the manufacturing and manufacturing authorization holders. They get information not only from India, they get information from other countries. So you need to get that information from other countries, discuss it in the India AEFI committee, and also the ITSU, Immunization Technical Support Unit, the CDSCO, and then you may need to make the decision based on that. And also, as again, one more important thing which struck me when Dr. Meera talked to us also, if you look at a country like India, you have such a heterogeneous population. You have people, in the south, you have people who belong to who have got certain characteristics. Some of them, for instance, have got certain unique features, the genotype and the phenotype. And also, for instance, they belong to certain nutritional status, diabetic status. So many things are there. So we need to find out whether there is a pattern of AEFI occurring in these unique populations. And it's important to determine that. Now, how do we do that? You need to prepare, each level, whether it is going to be a district level or a state level or a national level, you need to prepare consolidate your reports and prepare your monthly report and quarterly reports and annual reports and share the same. Maybe you may not want to share with WHO, but at least share it with India, with the national, people, and also with partners and to JRF. Why I am saying this is, I was actually impressed, very impressed, because recently I saw that earlier there was a publication of about 10 GBS cases which came in from an institution in Kerala. I think it was from Kottayam, where they had actually done a study at the Department of Neurology, or from a private hospital. They had actually done some work on GBS related to this AstraZeneca vaccine, which was really published. It really came out, and then we contacted them and also we got feedback from them. It's also important to share this and also publish this information, not only in your website, maybe in big publications as well. So I'll stop there because I know that I have only 30 minutes. Thank you very much.
Thank you very much, sir. We will take up the questions which is related to your topic at the end of the session, sir. I gracefully thank you for being part of the session, sir, and it was a very wonderful talk. We move on with our next speaker of the day, Dr. Chetak. Dr. Chetak KB, he is an assistant professor working in the Department of Pediatrics at JSS Medical College and Hospital. He has been involved in corporate hospital, after which he joined back to the JSS Medical College in 2012. His area of interest are nutrition, vaccination, and intensive care. He is a nodal officer for COVID-19 vaccine program at JSS Hospital, and he has worked in professional organization under various capacities from treasurer to secretary. He has 16 publications which are in peer-reviewed journals. With this brief introduction, I welcome Dr. Chetak, sir, and I request you to deliver the talk, sir.
Am I audible?
Yes, sir, you are audible, sir.
Screen is visible, is it?
Yes, sir. So please put it in the presentation mode, sir. Yes, sir. Thank you, sir.
At the outset, I would like to thank Dr. Ramesh, Dr. Junie Sebastian, and Dr. Ravi, sir, for giving the opportunity in this event. So it's a very difficult task after two stalwarts have spoken about the AFI. I'll try to do justice with my talk with the experience of COVID-19 vaccine surveillance and AFA report at our center. So as the previous speakers have already mentioned about it, I'll not go into details. So briefly, we started the COVID-19 vaccination program January 16, 2021, after the regulatory approval from the Government of India. Why we want to know the AFI? Because the need of the hour was, it was many healthcare workers, though the vaccination program was introduced for the healthcare workers, there are a lot of apprehensions in the public, in the healthcare workers. Should we go for a vaccine which has been done in a short span of time within 1 year? Many are, because when we started this program in our hospital, the response rate was 50%. And the rest, we tried to convince them so that it is safe and effective. However, we could increase to 60% by the end of 3 months. Then later on, as they enrolled and evolved and the public was involved, then the vaccination percentage came. So we had initially only the fact sheet from the vaccine, the Covaxin and the Covishield. The commonest from the Covishield fact sheet was tenderness, pain, warmth, redness. For any other vaccine, it was the same for both Covishield and Covaxin. The commonest of the Covishield side effects was lump at the injection site, fever, vomiting, flu-like symptoms. What we commonly saw in our setup was most of the first 1 or 2 days, the very common symptoms were commonly reported, and anxiety was also commonly reported at our sites. So these were the commonest which we saw in the first when we started the vaccination program day 1 or 2. The uncommon were dizziness, reduced appetite, abdominal pain, enlarged nodes, and excessive sweating. Coming to Covaxin, the injection site pain, headache, fever, malaise, nausea, vomiting, and rashes. Comparison between Covishield and Covaxin, the number of side effects in the fact sheets was relatively less in Covaxin compared to Covishield. The importance of this AFib surveillance was for early detection, investigation, analysis of the adverse event following immunizations. So, with the objective of the study to know the adverse event following vaccination, COVID-19 vaccination at our hospital site. So that the people who visited or took vaccination at JSS were included in the study. It was an active surveillance. It was started from January 13, 2021 till Jan 13, 2022 were included. And all people who took vaccination more than 18 years of age group are included in this study. So the inclusion criteria, population of any gender more than 18 years who received any dose of COVID vaccination irrespective of Covishield or Covaxin. At our center, we didn't have other vaccines other than Covishield and Covaxin. Exclusion criteria, population who were unwilling to participate in the event or were unable to contact of 2 attempts of telephonic conversations. This is the methodology. Subjects are provided with the patient information leaflet at the time of vaccination. Then we are subjected to collection of beneficiary details from the vaccination area on daily basis. Telephonic contact was made on day post-vaccination, and subsequently at 28 days after the vaccination, if they to visit for the booster dose of the vaccination, the follow-up was done there, or 28 days from the second dose, the contact was made. The analysis of the reports were collected for the severity, type, and causality, and the details were computerized for further analysis. So there are a total of 13,119 beneficiaries in the study period, both Covishield and Covaxin combined. But the people who were enrolled in the study for reporting AVFI was 10,662, which have accounted for 80.8%. The reason for others' exclusion was unable to contact after 2 telephonic contacts or unwilling to respond or confidentiality issues. These are the parameters of Covishield and Covaxin. There are 8,452 beneficiaries in the Covishield group compared to 2,210 in the Covaxin group. Male accounted for 3,934 in the Covishield groups, or compared to Covaxin 1,140. And the female 4,580 in the Covishield groups and 1,070 in the Covaxin groups. Now, many will be thinking, why there is a difference between the Covishield and Covaxin group? The main difference was, the reason for the difference in these 2 shield vaccination was, Covishield was started in January 2022. 16 January 2022, whereas Covaxin was approved in 13 March 2022. So the first 2 to 3 months, many people, healthcare workers particularly, were initiated the vaccine and followed by elderly people. The second reason was the cost. The cost of the Covishield was 780 rupees versus Covaxin 1,400 rupees. So many people hesitated. This is the practical reasons what we encountered or saw when people used to come for vaccination. Then Covaxin was not initially approved for international travel, so many people who wanted to travel, or the students who wanted to go for abroad for further studies, were waiting for Covishield rather than for Covaxin. Then the other practical problems what we encountered was Covaxin came in a 20-dose vial compared to Covishield as a 10-dose vial. The instruction was, unless there are 20 people, we are not supposed to open a Covaxin vial. So the end of the day, when the 2:30 or 3 p.m. the vaccination end by 4 p.m. There are no beneficiaries for Covaxin. Probably instead of coming tomorrow, they used to go for Covishield rather than Covaxin. And the availability of the Covaxin, which was not consistent, because sometimes we used to get this vaccination from the district health office. So they used to say there is no shortage of Covaxin, probably you have to promote more Covishield because of the non-availability of the Covaxin. So these are the reasons which we felt was one of the factor where the difference in the numbers between Covishield and Covaxin. What are the adverse events following immunizations? Total of 1,347 adverse events were reported. And the study population who developed AAFI was 886. The incidence rate was 8.3%. The average was 1:6 AAFI per person. These are the temporal association with Covishield, the first dose, second dose, and the booster dose. In the first dose, what was important observation, what I found out was the adverse event was common, was same, not common, was same in the between the first dose and the booster dose. However, the second dose, the adverse event was relatively less. Most of the adverse event happened within 24 to 48 hours. However, there was few incidents reported even up to 96 hours. These are the adverse event which was within the fact sheet was provided by the manufacturer. The commonest being the body pain and fever, followed by neck pain, tenderness, pain, redness at the injection site, swelling, headache, fatigue, excessive sweating, skin rash, feeling dizziness, flu-like symptoms, joint pain, being sick, enlarged lymph nodes, and abdominal pain. These are these AFI which we observed, which was not in the fact sheets. Tachycardia, tremors, lip swelling, insomnia, eye burning sensation, tingling and numbness of the digits, chest tightness, anxiousness, edema of the upper and lower limbs, edema of the face, loose stools, stomatitis, puffy eye, prickling pain on the leg, sneezing, body heaviness, reddish lesion on the foot, encephalopathy requiring admission, adenopathy, left arm pain, injection abscess, heavy headedness, eye irritations, breathing difficulty. The three serious other or autoimmune arthritis, autoimmune hepatitis, and leukocytoclastic vasculitis. Coming to the temporal associations between the Covaxin, the first and second dose data is available. The third dose, there was hardly any few requirement or the beneficiaries who came for third dose. One, because the third dose was approved only for healthcare workers, and the number of healthcare workers who were willing for booster dose was less. And the second thing was we were the 20 dose vial was available. So hardly few people were there for the booster dose of Covaxin at our center. And because of the regulatory guidelines that we are not supposed to open a vial unless there are 20 beneficiaries, we could not daily open a vial of Covaxin. So many people, healthcare workers, took the vaccination at the government centers. That's the reason we don't have the booster dose details of the Covaxin at our site. Coming to this severe the adverse event following the first and second dose, similarly like the Covishield, the most of the reactions happened within the first 24 hours. In contrast to the number of adverse events after 24 hours, relatively less in both the first dose and second dose. This is one change or difference what I observed within the Covaxin and Covishield. The number of adverse event following vaccination after 24 hours were relatively very low in Covaxin compared to Covishield. These are the fact sheets, adverse event from the manufacturer. The commonest being the injection site pain, swelling, redness and itching, headache, fever, malaise, nausea, vomiting, skin rash and rashes. The other adverse event which we observed was black spot on the left eye, which was not, we are not able to confirm it. It was an accidental thing or we could not find an association with it. Heavy headedness, boils on the scalp, pain in the hand, allergy, swelling, increased respiratory heart rate, and feeling tired. Relatively compared to Covishield, the number of adverse events were less in Covax. These are the severity of the AAV5, mild, moderate, and severe. One thing what we noticed was the adverse event of mild to moderate was more in a booster dose of Covishield compared to the first and second doses. we didn't have much beneficiaries in the Covaxin, so we don't have a data for the booster dose reactions of AFI for Covaxin. There were 6 serious AFI reported at our center. All these were for Covishield. One was leukocytoclastic vasculitis, which was even reported by Dr. Juni and the team. Autoimmune hepatitis, autoimmune arthritis, encephalopathy, injection abscess, and anaphylaxis. None of the beneficiaries at our site who received vaccination died following vaccination. This is a data of the AFI Mysore district collected from the DHO and the AFI center in Mysore. In Mysore, just for comparison, I put this slide. Total of 25,26,000 people have received vaccination. This is for more than 18 years. Both includes Covishield and Covaxin. The serious AFI cases are 12, which is yet to be, which are escalated to the state and where the causality association has not been described or approved. However, we should note this: there are only 2 deaths, and the association due to vaccine is not yet been declared. So, other serious AFIs has been discussed and has been defined. Whereas only 12 AFI has been escalated to the state, and the state has not taken a final call. The data is not available in the districts. However, there are only 2 deaths associated with vaccine of Covishield and Covaxin, for which the final association has not yet been declared by the state. So these are the type of AFI. One, vaccine product related information in the Covishield was relatively common compared to Covaxin. Vaccine quality defect reaction were none. Immunization related error was 2 in the Covishield. Anxiety related event was common in both, relatively small, and coincidental event was common in both the group. However, the coincidental event are more of patients, the beneficiaries becoming positive due to COVID vaccine following COVID vaccination. This was observed during the 3rd wave, where the booster dose was announced and many beneficiaries took the vaccine, and within 1 week they used to develop COVID disease. Then coming to the causality assessment category, consistent with causal association was common with both the Covishield and Covaxin. There is 1,000 cases in Covishield compared to 98 in Covaxin. Indeterminate for 11 and 6, and inconsistent with causal association was 196 in the Covishield and 5 in the Covaxin group. So these are the related risk group. What we found was the male gender relatively more adverse event compared to the female, and younger population had more adverse event following immunization compared to older children, and even the comorbidities did not prove a consistent association. What are the barriers with respect to the data collection? One was the telephonic call. There was non-response, or there were invalid numbers given, or inability to spare time by the beneficiaries to share their experiences or their adverse event. Difficulty to get all the data from the telephone calls. Mild AFI was not reported due to lack of understanding of the AFI. This was common even with our healthcare workers. Many of them are healthcare workers, and the mild AFI like fever, pain, swelling were not reported even by our healthcare workers. Then confidentiality, that not willing to disclose the information, or other barriers, why the response rate was 80%. So, coming to the conclusion, vaccination has been regarded as a major tool by many healthcare authorities and the communities for the achievement of public health success rate. Rarely serious events have occurred during the study periods, which reveal that Covishield and Covaxin have generally favorable safety profile. The vaccine safety studies help the healthcare professionals and general population realize that COVID-19 vaccines currently used in the countries are safe. Similar studies help to convince the population who are resistant to vaccination due to the safety issues and help achieve herd immunity against COVID-19. This, as speaking with the district immunization officer, the booster dose as of now in Mysore district is only 30%. There is still hesitancy among the population to take the booster doses, and continued AFI surveillance for ongoing better booster dose vaccination, including the pediatric population, is essential. Coming to the last slide about the word of the pediatric vaccination, though pediatric vaccine was approved between 16 to 18 group and Covaxin was included. There are hardly beneficiaries between the less than 18 age group at our center because the district authorities were directly reaching the schools and vaccinating there in a day or two. And hence we didn't have any pediatric vaccination data in lowest form. This is the state data, sorry, Mysore district data which is available. 15 to 17 years age group, 1,12,000 beneficiaries have received Covaxin, and between 12 to 14 years age group, Corbevax, there are 72,000 beneficiaries received. There was only one serious AFA reported, that is one case of GB syndrome in Covaxin group and none in the Corbevax groups. What I mean to say is, in the pediatric age group, the vaccine safety is very, very good as per the data available in the Mysore district. This one GB syndrome which was reported is an old case of GB syndrome. I attended the AFI meeting the last month. This particular child has known case of GB syndrome, treated 3 to 4 months back with IV immunoglobulin, and he was advised not to take vaccination. The parents are advised not to give vaccination for the next 1 year. However, the child was not aware. This vaccination program which happened in the school, he took the vaccine and again he developed GB syndrome. This is the only one AFA which has been reported in the pediatric age group in the district of Mysore. Thank you.
Thank you very much, sir. We are at the end of the whole session for today's webinar. Now I would like to take up some questions. Participants, if they have any kind of questions asked pertaining to any topic which was dealt today, I request you to please post it in the question answer session. For now, there are 2 questions which are available. This is for Meera Desai, ma'am. Ma'am, the question is, can the participants of phase 1 and 2 trials be included for phase 3 trial also? Ma'am, unmute yourself.
Oh, sorry. Yes, Shilpa. There is no clear answer and any rules or regulation that you can include or you cannot include. However, in the best interest of your data collection, best interest of the subject safety, better to avoid it. One more thing, study 1, phase 1 will be done at the CRO in the hospital setup, while phase 3 is done in the community based. So obviously there is a difference. So better to avoid this. Hello.
Okay, ma'am. Okay, ma'am. We have one more question to you. Question is like many breakthrough infections, even after 3rd dose of corona vaccine, have been reported worldwide. How can we address this question of effectiveness of COVID-19 vaccine in real world?
Okay. The effectiveness, when we talk about the efficacy and effectiveness, these are 2 different things. Most of the clinical trial, the reported efficacy and what the regulators also look at it, that if it is somewhere around 50 to 60% protection, that is fine. The other way, the other endpoints in clinical research, which are also considered, that reducing the severity of infection, reducing hospitalization, and reducing the death rate. These are the another separate endpoints for efficacy point of view. Now, when the vaccine is approved, comes into the market, and thousands of general people are taking it, that time it becomes an effectiveness to find out to what extent it is really effective in preventing the infection, reducing the severity. Hardly it will happen that, especially with the corona vaccine, that it gives 100% efficacy. So what needs to be done, if we are really interested, we can take up further studies with more population, and the AEFI data, which is already mentioned by Dr. Juni and Dr. Ram, that whether such type of adverse events which are reported, we need to go back, find out the details from these reports which are captured in our database, that what number of doses, the booster dose, 3 dose, 1 dose, how many doses were taken. That is another way of doing it. Whether the protection is there, to what extent is able to protect. Suppose in the community, 100% vaccination is done. Out of that, how many number of patients they really had these events after 2 doses or 3 doses? That is another way of taking up the research. It's a very good area and that can be done now. But we don't expect, neither the regulators expect that it will give 100% protection, because such rare type of adverse event will be reported only once the vaccine is approved, available in market, and you need to have a large database. You need to have epidemiological studies now. That is the way you can take it up. I hope I'm able to answer this question.
Yes, ma'am. Yes, ma'am. Thank you so much. They have maybe other panelists also can join us. The question is, any information on long-term complications after vaccination? If anybody, Sir, Madhav sir or Chetak sir?
Thank you. So, you know what, as Dr. Meera was just mentioning now, many of these things we are also slowly beginning to discover also. To be very frank, for instance, when the clinical trials went on when the AstraZeneca vaccine was going on in the UK, when it was done by the Oxford AstraZeneca vaccine, at that point in time, we were getting information about transverse myelitis. And at that time, we had really not heard much about this TTS, the thrombosis thrombocytopenia syndrome. So we did not know that. But once the vaccine entered into the market and once it started getting used, that is when, we started getting clusters of these cases where people were finding more amount of clotting disorders which were kind of manifested. So in other words, only especially with new vaccines, this becomes a problem. But when you look at existing vaccines, like if you look at your pentavalent vaccine, your BCG vaccine, polio vaccine, what I mean polio vaccine, I'm talking about the trivalent polio vaccine or the IPV, and not about the NOPV type 2 and things like that. Now, those kind of traditional vaccines, we do have long-term information. But when you look at newer vaccines like the COVID vaccine, the malaria vaccine, the dengue vaccine and all that, only when you start using it, we will be able to have this information. Over.
Thank you, sir. Junie, I think you can answer for them. If the symptom is just vaccine site pain for 1 day and recovered, shall we report that?
Yeah, we can report the adverse event. So it may not need a further investigation, or it do not falls under the category of serious. So it can be reported in the weekly report or the monthly report. But to the pharmacovigilance program of India, we can directly report it. I do not want to go through the district immunization office, but all these kind of reports the events need to be reported to improve the reporting culture which we have.
Yeah, that's right, Junie. There is a question basically based on the vaccine production. Participants were keen to understand if there is a bias. That means it could be made unintentionally or unintentionally. How would we validate the safety efficacy data available? So, Meera ma'am.
Yes, what is the question? Sorry, what is the question? In the process of?
Yeah. In the process of acceleration of vaccine production, there would be many instances of bias that will be undertaken intentionally or unintentionally. How do we validate the safety efficacy data available?
Okay, what do you really mean by the? I would like to ask question to the person who has proposed this. What do you really mean by bias here? The production of vaccine is accelerated. right? And that is the job of the manufacturer and marketing authorization holder. How the bias will come in? You mean the bias from the reporter or the patients, or who will have the bias? The question is not clear. What is the relationship between, see, the bias is usually considered when the clinical trial is going on. Once the vaccine is already in the market, the bias from the sponsor, from the investigator, from the, I would say, a clinical trial people, I think that factor will not come in. We need to understand is what are you trying to ask? Is it the patient bias? The patient will be biased?
They have not really said about it. Maybe, ma'am, during the production process, maybe they are saying that because the number of the demand is too high. During the process of vaccine production, there could be some, maybe they are saying negligence, I think. So they are basically asking about the safety efficacy data.
The safety efficacy data will be generated in clinical trial when the process of research and development is going on. And if he's asking, the person who is asking this, if he's asking about the error which is taking place at the large scale production, then it is the responsibility of the regulatory authority to, and as well as the state regulatory authority to continuously monitor the production site, frequently inspect the site as well as the manufacturing process, whether they are meeting the standards or not.
Yes, ma'am. Yes, ma'am. Madhav sir, if you could just let us know the participants is for how many years monitoring of AEFI of COVID vaccines will be undertaken?
As long as the vaccine is used, we'll have to have monitoring of the safety of the, the COVID vaccines. Because we cannot stop. Any vaccine, which for that matter, which is used, it will be post-marketing surveillance will be conducted. There is no question of stopping it at any point in time. Over.
Sir, I have a question for Dr. Madhav.
Sir?
Many students and healthcare workers come and ask me about the safety of booster dose of Covaxin. One. Second thing, following many students travel abroad from here, they visit America, and there the vaccine offered for booster dose is different. So what is the data which is available as with WHO about the safety of booster doses from other countries?
Right, okay. Now, what we do is, there is actually the global database, which is actually called the VG base. And people in the PVPI, the Pharmacovigilance Program of India, they have access to this particular database with only one person, because as I said, this particular data is so confidential that it is not even shared with, even within WHO itself, only very few people have got access to this particular data because it should not be unauthorized. So if you look into this particular data, in fact, each country has the right to look into this data and give you this particular information. So for different vaccines, it's actually being carefully monitored. I just can't tell you offhand with statistics and things like that, but then I can probably get back to you on this and provide you with more information. Thank you.
Second question, is the EFA reported is generally the same across all the products of vaccines available in the countries, or in few areas you find some vaccines has more complications compared to other vaccines? The general kind of, not without naming the vaccine.
Thanks. You know what? It also depends on the quality of the surveillance system, the capacity of the country to do causality assessments, and also from our point of view, when you look at the data, honestly, the data, for instance, why people keep on asking us, why do you trust the data coming from the United States, for instance? Why is it? Honestly, it's very good. It is very robust. The approach, they have got a system called the VAERS, Vaccine Adverse Events Reporting System. And then it is so systematically reviewed and then they publish it in their public domain. For instance, there is something called the ACIP, Advisory Committee on Immunization Practices. And then those kind of decisions are available in the public domain. So you see what I mean? Whereas if you look at other countries, even in Europe, for instance, if you look at the MHRA, the one which is there in the UK, it is much better than what you see, for instance, in France or in other countries, because the reason is there is a systematic approach in following up and publishing it and making it available in the public domain. You see what I mean? So it is very heterogeneous.
Thank you, sir.
Thank you, sir. Chetak sir, you have a question. Participants wants to understand, is there any case of drug-vaccine interaction or case in which drug or disease can interference, or for example, they are asking rheumatic drug interference in effects?
Yeah, this initially there was recommendation from the American Academy Rheumatology Association for patients who receive vaccines. They told, for example, anti-rheumatic drugs, there could be cyclosporine or methotrexate or any immunomodulated drugs. 15 days the drug has to be stopped, and then the vaccination was to be taken because they didn't have any data on it. Subsequently, there is no recommendation from the Rheumatology Association, because the data what I'm sharing is only from the recommendation of the American Rheumatology Association. There are no recommendations from other rheumatological associations. So as of now, there is no data to say that these drugs, those patients on any of their drugs should have any interference or effect on the vaccines. What we followed was any patient, irrespective of the status, including the medication for HIV or tuberculosis or immunosuppressive drugs, the patients can take vaccines. And we did not find any associations or more people who were the drugs intake and more serious adverse event. There was no observation as per the study or the data which is available from JSS. I think Dr. Ram could elaborate his experience.
Yeah, thank you. Actually, if you just see, if I remember right, what you said is absolutely true. Absolutely true. Dr. Chetak, absolutely true. The thing is, when we were writing this manual on causality assessment of AEFIs, there is one particular question which is asked during causality assessment. Do you think that this one could be a product of a drug? We have, when you do this assessment, you will find that many of the times the reports of adverse events which get reported might be probably coincidental, but trying to establish that connection between the vaccine and the drug is actually extremely challenging. Because what we need to do is we need to do prospective clinical trials, and then sometimes it is unethical to do that in field situations. This is what is a problem which we face. But I fully agree with what you said.
Can I ask Dr. Ram one question?
Yes, please, Dr. Mira, please go ahead.
Are you coming across any adverse events reported due to RNA vaccine, mRNA vaccine?
Lots.
Okay.
The thing is, you know what? You can also ask yourself, do you have this information? The answer is yes. And is it available in the public domain? Partially, yes. The reason is if you want, you can just type it down. There is something called VIGI-Access, VIGI-ACCESS. So in the public, because as I said, this particular data is very confidential. We cannot really share it with people. But if you're really interested, you can actually send a letter to the PVPI people in India, and they will be able to access this VIGI-LICE data, and they'll be able to give you an exact answer with numbers.
Yes. So that VIGI-Access is for the public. Everybody
Correct.
can access it. But VIGI-LICE is only to the national centers. They are only to the national centers. Because mRNA vaccine, it is proposed that it will be having more efficacy and it will be very safe. That is proposed. And even you can produce thousands of doses in no time.
But that is true.
The protein sequence. This is all, I don't know whether it is hyped or it is really true.
It is true. It is true. What they have mentioned is absolutely true.
Okay.
But the reason is people come to us and ask us exactly the same questions, Dr. Meena. Why do you want to see about, because you know what, Especially, I know the background with which many of these questions are being asked, because some countries might be manufacturing a viral vector vaccine, another country might be manufacturing an mRNA vaccine, and each country wants to promote its products. This is what really happens. Now, if you ask me, the incidence of pericarditis and myocarditis with mRNA vaccines are definitely there. The connection has been established, and it has been established with the second dose of the mRNA vaccines. And with Moderna, it is confirmed. Okay, so it's not that it is not not confirmed. Okay, and also there have been reports of sudden deaths which have occurred after mRNA vaccines also. But then, if you ask me again on look at the other side. So this is when you look at the flip side. When you look at vaccines like the viral vector vaccines, we have had confirmed cases of thrombocytopenia syndrome which have been reported from several countries. Now, just because honestly India doesn't report, it does not mean that India doesn't have. It probably means that the surveillance system is not picking it up, or it is picking it up, but it is not reporting. You understand what I mean? So, because again, there must be a question about case definitions. People must be aware, they must be sensitive. Even for instance, if the time interval between vaccination and the occurrence of the TTS is somewhere between 3 to 30 days with a median time of 9 days. You understand what I mean? Now, if it is coming up within the 30th day, and if the treating physician does not connect the vaccination to this, it will not get reported.
But there was also connection between the corona infection and myocarditis.
Absolutely.
And
in fact, there is,
so it is extremely difficult to whether it is the infection, because people here, like I'm from Gujarat, people in Gujarat are getting the infection after maybe 6 months, getting cardiac events after 6 months, 1 year. So they are trying to relate to the original pathogenesis of corona rather than to the vaccine.
It is very difficult to differentiate that.
Very difficult, yes.
Yeah,
true.
and as you said, when you look at it, the incidence we are looking at the benefit risk ratio. If you look at the MHRA website, they have done a beautiful analysis of TTS with COVID versus this one. It is something like 200 times more if you get COVID causing thrombocytopenia syndrome compared to the vaccine. So it's a question of risk benefit. We are not guaranteeing, as you said, nobody can guarantee. But at the end of the day, you need to choose between the vaccine or the disease. So that is what it is.
Moreover, the number of cases, the number of people vaccinated, and out of that, how many are getting. So even the large epidemiological studies will be able to tell us. And everybody is now interested with corona vaccine. So I think the time is the solution. We need to wait and collect the data, and we need to have a very proper infrastructure and the reporting system. That is, I think, and a complete assessment. That is what is required.
You hit the nail on the head, Dr. Meera. Because honestly, the thing is, if you look at what really happened for us, the TTS cases and all that stuff actually did not come from developing countries. It was actually reported from the Nordic countries, like, Sweden, Norway, and those kind of countries, even though the vaccine was used all over the world. And then it has been confirmed that there is a connection between the thrombocytopenia syndrome and the viral vector vaccines. It has been confirmed.
Okay. Thank you. Thank you.
Thank you so much, ma'am and sir. Now I let you take the opportunity to present a vote of thanks. Today we have witnessed a very important session of vaccine safety monitoring and research. First of all, I would thank our management at JSSCHR for giving us a support in conducting this particular webinar. I thank the Regional Training Center, PVPI, and also the members of the Medical College and Hospital. I thank our key important persons who are involved here. Dr. Jai Prakash, who is a Senior Principal Scientific Officer from PVPI, though we could not really make his talk very clearly, but he was expressing his gratitude in organizing this particular talk, and as well as he was wishing all the speakers for being part of this, and he could say these words as well. So I thank him for joining us at this event. I thank Dr. M.D. Ravi sir who has been always a support and a main key person in vaccine safety at the hospital. I thank you very much, sir. I thank Dr. Meera Desai, ma'am. She has been really with us from the starting till the end, and you have patiently answered many questions. Thank you so much, ma'am. Thank you so much. Dr. Madhav Ram, sir, I really appreciate, sir, because even apart from being the time difference at Switzerland as well as in India, you could be with us with your busy schedule, and you have shared all your expertise in this particular field of vaccine safety. Of course, your words and your talk would really be very informative. And thank you so much, sir, for being with us till the end and answering all the questions. Thank you very much.
Thank you.
And
Thank you.
I take the opportunity to thank Dr. Chetak, sir, also. Sir, thank you very much, sir. With your busy schedule, you have made your possibility to be with us and also take part in this webinar. Dr. Julie Sebastian, thank you, Dr. Julie Sebastian, for being part of this. And your information has been dissipated well. So thank you so much for being part and also being as the organizing person for this particular webinar. Dr. M. Ramesh, sir, a coordinator of AMC at JSS Medical College and Hospital. Thank you, sir, for supporting as well as motivating in making this webinar happen from the starting from the organizing till the process. You have been a support, and thank you so much, sir, for being. Because of his medical emergency, he would not join at the end of the session, but he has given his greetings and thanks to each speaker and the organizers, especially present here with this webinar. Thank you so much, sir. I also thank our principal, sir, Dr. T.M. Pramod Kumar, sir, who has, though he is not physically present here, but he has been supportive and he's given his full support in organizing and conducting this webinar. I thank the IT people who have given us the platform to make this webinar happen with a smooth conduct. I thank Dr. Balaji also, and all the participants who have presented here and they have actively participated in the webinar. I thank you, each one of you. Thank you so much for being part of this.
Bye.
Yeah. Thank you. Thank you very much.
Thank you.
Bye-bye.
Thank you. Bye. Bye.
Thank you, sir. Thank you, madam.